That one actually was sent. I have it downloaded already.
Okay. All right. So we will begin with some introductions. I know that we already talked a little bit, but I always like to ask about your experience, about your expectations about this training, mostly because sometimes you've used Columbus or not, and that we'll talk a lot about your experience and expectations with Signals Image Artist. So I would like each one of you to introduce yourself. I know that we have some people that is not here in the call, but is on there with you. So if you can just go ahead and start some introductions. I'm going to start with you, Beverly.
Great. Yeah, thanks so much. I'm Beverly. I manage the Chemical Screening Center at UC Santa Cruz, which is the facility that runs the Signals Image Artist software that we're here to learn about today. We also have the PerkinElmer Phoenix Plus instrument that collects data in the Harmony software that we're going to be analyzing in Signals. And my facility also has an Image Express imager that has the NBC Store Tools database of data that we're also interested in analyzing in our Signals package. And most of the people in this room on our side are users that I've trained. If you need any more training on Harmony or the Phoenix or the Image Express, please come to me.
Okay, perfect. Thank you. All right, next one.
Hi, I'm Matilda. I am working in screening center and I'm shared between microbiology lab and this one screening center. But in both places, I will be doing high throughput microscopy. So I'm interested to use this software to analyze my data from the biofilm which bacteria create and also cell culture stain with different stains.
Okay. Yep. All right. Awesome. Thank you. Okay. Next one.
Not sure.
Yes, I'm not sure. I do a bunch of data things. I've mainly worked with MetaExpress before this, but so far the UI seems to be very similar to what I've done before. So hopefully it should be fast. But haven't touched anything from Phoenix, or sorry, from Signals, I should say, or Columbus.
All right, perfect. Thank you. Is that only you three, or do we have someone else with you, Beverly?
I think Sahar might be here.
No, she left.
Oh, she left. Okay.
Okay.
And Oswald, is Oswald not here? He said he was coming.
No, I think he may have forgotten. I can go poke him if you want me to, but I think he has other things to work on.
Yeah, no, it's okay. He hasn't yet used Harmony, so maybe he's not going to be getting Signals much either.
All right. Okay, thank you, everyone, for your introduction. Let's continue here with the objectives of this training. Mainly is to become familiar with Signals Image Artist interface, perform image analysis with high content images, and we will enable you with the tools to run this kind of analysis. We will also learn how to run a batch analysis on some plates. So there are some tools that we will go over here in the training, so you can apply that on your daily work. The activities of this training, first we will have a lecture with the PowerPoint presentation. So there are some concepts, topics that I will explain using the PowerPoint presentation, but we also have some hands-on activities. So here is where you will use the sample data that I sent, so you can follow with me during the training and play with the application with the sample data. And at any moment, you can ask any questions. I will be doing some pauses between topics so you can ask your questions. But if at any point you have any question, feel free to unmute yourself, raise your hand, use the chat, and we can cover the question at the moment. Okay.
Great.
About
That's
expectations.
reminding me of myself.
Yeah. Sorry.
No, that's all. Thank you.
Okay. About expectations, your participation is key for this session. Feel free to ask any questions. You can share your screen if we need to clarify anything. Minimize your distractions, mute your cell phone, mute your email. We will have some breaks during the training, so you can do all of those activities. And about your questions, I will do my best to answer all of them, but some may require a follow-up. If I don't have the answer to a question right now, I will always do an email follow-up with you after I do some research. All right, these are the topics that we will cover today. First, well, we have introductions in the agenda review, which is right now. Then we are going to cover the support options, so how you can get help from support while using Signals Image Artist. Then we will explore the user interface, so what are some key elements. Oh, sorry, something happened with my monitor. Just let me share back again my screen. Okay, just 1 second. Okay. Yeah, so we will explore the user interface, how, well, the different icons, menus, elements that we will find on there. Also, the help guide. So what is the official documentation of Signals Image Artist if you need to answer any questions? And then we have the Image Artist workflow. We will begin with the browse, so how you can browse for your data. We have import export, which is basically what we just saw with Beverly's screen, how to import the data using Simaloader, and we also have the option to export. Then we have the image analysis. How can we build a basic image analysis sequence using building blocks? And then we have the batch analysis. So once you have an image analysis sequence, we will learn how we can run that sequence in bulk in a whole plate, for example. And then at the end, we have an advanced image analysis topic. So here we are going to talk about how you can analyze texture, morphology, and how you can use the Phenologic, which is a machine learning algorithm that we can use to select population and classify cells. At the end, we have a closing with final questions. And also, I will send to you a survey so you can provide feedback about this training session. All right, so let's begin with the first topic of today, which is the support and how you can get assistance. So these are some resources that we have available to you. We have the PerkinElmer website. We have some webinars in LinkedIn, so you can always check LinkedIn and see some webinars that we have available. We have a YouTube channel with some videos of Signals Image Artist and also other PerkinElmer products. We also have the Signals Image Artist help, so that is the official documentation of the software. And then we have the PerkinElmer Informatics support page. So in the PerkinElmer Informatics support page, you will find some other resources like the FAQs, knowledge base, support news. So this is an open community where you can read about frequently asked questions. We have knowledge base articles and support news if something is going on with Signals Image Artist or with any other PerkinElmer product. Support will post in here any news. And if you have all of these resources and you still have questions or have issues, we have the support team that is available to you. So if you have any question, you can contact support and you can submit a ticket with them here in this page, and I will show you that in a moment. Once we go to the system, you can click on contact support, fill out a form, and a support technician will get in touch with you by email, so they can help you troubleshoot any issues or questions that you may have. Another way to contact support is using this email, informatics.support@perkinelmer.com. In here, you can directly contact support as well. Okay, and then we have the CIMA help. The CIMA help is the documentation, the guide for Signals Image Artist. So if you have any questions after this training or when you start using Signals Image Artist with your data, you can come here to the CIMA help and you will see information about all the different features that we have in the software. Okay. Now, about the user interface, well, as you know, this is a server-based application. It is accessible through web browser. Google Chrome is preferred. At the top of the user interface, we have the menu bar, okay, here at the upper right corner, where we can access job status, that we will see during this training how we can use the job status, the help, and also you can log out from the application here. At the left side, we can choose the user, because in here you can choose your user to see your data, or if you belong to a group with more users, you can see data from other users in here, so you can select from this dropdown a specific user and you can see the data from that user. We also have in here the primary navigation, this data tree that we will use to, let me put a pointer in here. This data tree in here that we will use to browse our data. In here we have screens, plates, measurements, and we will learn in a moment what is the hierarchy to search for specific data in this primary navigation. Okay. And at the left side, we'll also have the secondary navigation. So the secondary navigation is to select the specific wells or fields of a plate. So once you choose a plate in the primary navigation, in the secondary navigation, you can select a well or a field to see the image of that specific well or field. At the center, once you select a well or a field, you will have the image view. So here you can visualize your images, and below the image we have the table view. Here in the table view, we will see some image information when you select images, but if we select analysis, or if we select results or any other type of data that is in the primary navigation, here in the table view, we will see some different information about the results, about the analysis. While we're doing the image analysis sequence, in the table view, we will see some preview of the results. So the table view is very useful to review any kind of information of the data that we are displaying at the moment. Okay. All right, let me continue here just a moment. Okay. Next, we have the different levels that we can see in the primary navigation. So in the primary navigation, we have a screen, we have a plate, measurement, and we have results. So you can see here the level. The screen will be the highest level folder that will contain all the different types of data. Then inside of the screen, we have the plate, and then the plate can have multiple measurements. The measurement will always have a timestamp. Okay, so in here you can see then the results, the analysis, also assay definitions or plate layouts that are at the measurement level, and that can be, well, we can have multiple measurements for one plate. Okay, we can also have plate layouts globally, so that will be at the screen level. So here we can also have global assay definitions. Now here inside of the measurement, you will notice that there are some icons in here that we need to understand to identify the data. This icon that we have in here will be always for assay definition or plate layouts. The second icon in here is for results. So when you see this square blue icon, those are results that are coming from a previous analysis. And then this icon that we have in here, this green with blue, these are for analysis sequences. All right, so this is a sequence that you can later apply to in another plate or in another measurement and get results with that sequence. Okay. So we'll continue with the next one. Then we have the secondary navigation. So we know that a plate can have multiple wells, and a well can have multiple fields or planes. The fields will be positioned in a well, and planes can be images at a specific height in a stack, like slices of a well or a field. So the idea in here is that in the secondary navigation is that you can select multiple wells to create like a global image, or you can select just one well at a time, or you can select one or multiple fields at a time. So in the image view, you will see the global image or the individual image depending on what you choose on the secondary navigation. Okay. Then once you select an image, we have some image view settings. So the application will allow you to use some different visualization tools to zoom in, zoom out. You can change the coloring of the image. You can change the channels to highlight specific structures depending on the purpose of the analysis. And we have some other tools in here that we will explore in a moment with more detail. But you see here that we have this table with some drop downs, some checkboxes that we can use to change the way that we visualize a specific image depending on the purpose of your analysis. Okay, and then we have the table view. So as we said before, the table view will display any kind of data, detail, information about an image, about results, about an analysis sequence, and the information that is displayed in here will depend on what kind of data you choose. If it is a result, measurement, or analysis, then that information will change. Okay, good. So with that said, oh, sorry, I see that Beverly and Matilda cannot hear me. I'll just put something in the chat. Akshar, can you hear me, or is it a problem with Beverly
Yeah, I
and
can hear you.
Matilda? Okay.
If you want, I can get
Okay, can you hear me now? Okay, it says it's reconnecting, so let's give it a moment.
Can you hear us? I think we're good now.
Yes, okay, perfect, good. All right, so now that we have explained how you can do the primary navigation, secondary navigation, image settings, and table settings, let's go to Signals Image Artist and let's start with a few images on there so we can see this in the software. Okay, so I will be using a server that is different from yours. This is a test environment. Here I have the same sample data that I sent to you. This is the RMS, which in your case you have it as PerkinElmer sample data or the name that you put in there when you did the upload. I have some other images that I will be using for this demo because we will use the RMS in the next tasks of the training. So if you don't have the images that I will be using in here, that's okay. Don't worry. You can just watch the demonstration in here. You can follow along with any other images that you have available. And later we will do some tasks with the ready-made solutions that we have uploaded. Okay. All right. So first, let's recap some items in here in areas of the user interface. So we said that this will be called the menu bar, where you can see the job status. You can go to the help. You can also log out from here, which is very important to show is the help. So you see that if you hover over help, you will have the SIMA help, which is documentation of Signals Image Artists, technical support. So you can go to the PerkinElmer Informatics support page, and about SIMA is information about the software. So if we go to SIMA help, that will open a new tab with the Signals Image Artist help. So here you can read information about the software, right? Some key features that you can read more about it. It's a very useful resource once we finish the training, because if there are some things that we don't cover on the training, you can come here and read more about those if you have any questions. Then we have the technical support. This will take us to the PerkinElmer Informatics support page. So we have a knowledge base where you can read about knowledge articles. We have FAQs, so these are frequently asked questions. Any questions that other users have had in the past, you will find those in here, and the answers from PerkinElmer support. And then we have support news, right? These are for the support news, and FAQs is for any PerkinElmer product. So you need to always make sure that you filter by Signals Image Artist so you can see knowledge content and news about image artists only. And if you need to get in touch with support, you can click on this contact support button that is at the upper right corner. You will need to fill out a form. Oh, I think I have the wrong access here, but the idea is that if you click on contact support, you can fill out a form, and support then will get in touch with you to troubleshoot any issues or questions that you have. Okay. All right, let's go back to the user interface. Then here, if we go to the left side, we have drop down for the user. So as I said, if you belong to a same group, to a group with multiple users, all of you can see the data of the users of that specific group. So if you select all, you will see all the screens of the users that are part of this server. And you will notice, for example, that in here I have a folder that is gray color. This is telling me that this folder is not mine. This is from other users. And if I select my user that I'm using the admin, so I need to choose admin, then I will see all the data that is associated to my user, right? Since right now you don't have any groups, if I'm not wrong, then you will see only the data that you have on there individually. But if in the future you have any groups, then you can see data of other users.
Can I ask real quick about the
Sure.
nomenclature? Like, I'm confused why it's called screens. Like, is this supposed to mean like each folder is going to be for a different project or for a different experiment?
Yes, that's right, because the screen will contain multiple plates, right? And usually the plates belong to one project or to one experiment. And then the plates will have measurements. So you can have multiple measurements for one plate, or you can have just one measurement for one plate. But basically, yes, the screen will be like the folder with data of a specific project or a specific experiment.
So is the what is it called instead of screens when you're in Harmony? Or is this assigning it to different screens? Is that something you only do in Signals? Like I know the terminology like experiment and measurement, those we have on Harmony, but
Okay.
screens is just an level. So when at what point do you put it in a folder and call it a screen and name it?
I understand. Well, not sure how it is called in Harmony. I don't have experience with Harmony, only with Signals Image Artist, but I know that Arturo has some experience, so I can take that question. I can ask him, and I can follow up with you, Beverly. Let me just take a note here, so I remember to ask that question. You said that in Harmony you have something that is called experiments? Or is it just plates and measurement?
There's a thing that's, there's a thing called, was there a thing called experiment? There's definitely measurement, and then there's analysis.
Yeah, it's evaluation.
Evaluation,
Actual
yeah.
results.
Evaluation.
Is evaluation. The measurement is the images, and then the actual analysis sequence. But we tend to sort of sub in terminology from using MetaExpress, where images are images or acquisitions, and then measurements are literally the results from applying a set of image analysis a journal essentially, just the actual method.
Okay. Yeah,
I'm just wondering at what point in the workflow do we assign a screen a name and say, okay, these images are for that screen and these other images are for a different screen. Like say we're screening some botanicals and we're also screening some anti-inflammatory something, then are those different screens, and when do we like assign it to a screen? Or like if we have antimicrobial screen, that's hopefully in a whole different folder. Like at what point does that happen?
So I guess a related question: can we just have a single upload of the images so we don't bloat image artists too much, but then associate that same plate of images with multiple screens?
The screen in this case will be like the folder that is containing everything, right? So you mean like uploading data in an existing screen? Like an existing folder?
Like we have some reference data sets where the data that somebody collects, at the beginning then gets used by multiple other projects and other labs and other purposes. It's like a reference data set. So that's kind of like for all the screens, if we're understanding the word screen correctly.
Okay. All right. Okay, I will take that question and I will discuss it with Arturo, that I know he has experience with Harmony, to see what is like the how that is related with Harmony in that case. In Signals Image Artist, what I can tell you is that the measurement, I know that Axel mentioned about the measurement, that it's results mostly, but in Signals Image Artist, the measurement will contain the images or the data, the results, and the analysis sequence. So for example, you see here that we have this measurement, and inside of the measurement we have these results, right, that you can identify with these icons, and we have these 3 sequences of analysis. All right, and if I click on the measurement, it will display in here in my image view the images of that specific measurement. So you will find 3 types of data in the measurement. Okay, and that's basically how you can browse for the data, right? If you select a measurement, you will see the image, and then in the secondary navigation you can select a specific well or field that you want to view in the image view. If you want, you can see. Yep, go ahead.
For the folder, that's just a date. That's the measurement. Is
That's
that
right.
the date that the images were actually collected? Like the light passed through the sample and the camera collected the image. Like, is that the date when that happened?
Right. Yes. Is
Okay,
not the
great.
date when you upload it, is the date of the instrument. Okay. Here you can also see multiple wells or fields at the same time. So if you want to see all the wells of this specific measurement, then you can just right click, and in here we have select all wells if you want to see all of them, or we can click on begin multiple selection. All right. So if I selecting here all wells, right, it will have all the different 3 wells of the measurement, or I can click on begin multiple selection and select or deselect the specific wells that we want to see. And then we right click again and click on end multiple selection. Okay. Same for the fields, right? That is for wells. Right now, we are only looking at the field number 1 of a specific well. So in this case, I can just select this well, and then I do the same, select all fields. So now I will see all the fields of the well C3. Okay. And if I
A
select
quick
all the
question.
wells,
It looks like
yeah.
there were 3. It looked like there were 3 fields, but there's only 2 squares.
Yeah, that depends on the data that you upload, right? I don't have information about what was the instrument used for this data. It depends. Sometimes I know that when you uh, run the instrument, you can decide to capture images of specific wells or not all of them, right? So that could happen in here. Maybe we had 3 fields, but we decided to only visualize the field 1 and the field 3, and then the software will have in here only 2 fields because of that. But it will identify that there was something in the middle that we didn't capture with the instrument.
So yeah, that's why there's black images.
Right.
Okay, they're like empty. Because there's 3 by 2, and then if you look, it's actually 3 by 3.
That's right.
Okay.
Yeah. Maybe in the
Thank you.
instrument, they decided to not capture the image of the middle. Okay. All right, so that's how you can use the primary navigation and the secondary navigation to visualize the image here in the image view. Actually, your question about multiple sites, is that multiple wells and fields? Did that answer the question? Okay, good. All right, so I will select just one well and one field. So now we can talk about the image settings, right? So here we have some tools, controls, channels that we can use to change the way we visualize a specific image. In the tools, we will find first some zoom tools. So in here we have some zoom values that you can explore, right? And you will see the image in full image or a specific zoom value. Okay. Then we have this 3 options for pop-up zoom, and follow focus. The pop-up zoom is just a square, a little window that you have in here that if you just explore the image, you will have this square that will work as some kind of magnifying glass where you can see the structures zoom in. Okay, so that's the pop-up zoom. Then we have the follow zoom and the follow focus. This follow zoom and follow focus will work if you have this floating image, all right? The floating image is very useful because if you want, for example, to change the channels, change the coloring that we will see in a moment here, right? So we can edit the channels, edit the coloring, and you want to compare against the original image, we can use the floating image to do that, right? So somehow we can see the original image here in the floating one, and then at the in the background we will have the image that we are editing. So if we are using this floating image, we can use the follow zoom or the follow focus. The follow zoom is that any zoom that I do on one of the images will apply to the other image as well, right? So I'm just using the scroll in my mouse, and I can just scroll in, scroll out, and I will zoom in and zoom out in both images, right? Because the zoom will follow on both images. Now, then we have the follow focus. The difference between follow zoom and follow focus is that the focus is to follow the same structure or the same place of the image that you're looking right now. So if I use the follow focus, okay, if we compare here, you will notice that anywhere that I zoom in, I will see the same structures, right? If I move around here, I will have this cell, which is this other cell is the same. And if I move around the image, right, I will turn on here drag image so I can move around the image. It will follow to the same place of the image, right? So that's the difference between follow zoom and follow focus. Okay. I will exit this window. Then we have the viewfinder. In the viewfinder, you will have this window where you can see where you are located right now. So if you zoom in and zoom out, and if you move around the image, you will have this window that will help you to have a better idea of where you're located in the global image. Okay. Next, we have the measure, but to use the measure, we need to uncheck the drag image, right? Both options won't work at the same time, so I need to deselect drag image, and the measure will work for two things. You can click on a spot and you can see the coordinates and the intensity of each channel, or you can also make a rectangular area with the mouse and measure the pixels of a specific structure. So if I click on this spot, for example, I will see the coordinates, X and Y, and then we also have the intensity of each channel. Okay. And then if I do a rectangular area in here, it will give me the pixels or micrometers of this specific rectangular area. Okay, so that's for measure. The drag image, we already saw this, is if you zoom in, the drag image will help you to move around the image. Okay, and that's it for the tools. Then we have the controls. So in here first, we can change the coloring. So you can select one of the three display modes to view the image as required. We have highlight. It will show as many details as possible. Actually, that is the default that we have, the highlight. The enhanced is less intense. So if I select enhanced, I will see that the structures are less intense. In the standard, the intensity is not modified, so it shows less image details, right? So this will be the standard. I will go back to the highlight because in here it will show me as many details as possible. So that's what I prefer for my image. Okay. Then we have the show scale. So this is a scale that you can use to judge the size of the object. So for example, if I select bottom left, I will see here at the bottom left of the image a scale of 20 micrometers that I can use to judge the size of the objects of my image. Then we have the flat fill corrections. This can be selected only if it is available for the current measurement. It depends on the instrument and how you measure the data. If you use none, then the image is not corrected, and that is the default. We have the basic, that is to correct the background but not the foreground, and the advanced, that will be available only if the data will allow it, is to correct both background and foreground. Okay, so if I select here none or basic, I will see the difference in the background, the image in general. Okay. Then we have the sharpness. So this is a filter to enhance the sharpness of the image, right? So we can uncheck or check the box. Sometimes that will affect the image, sometimes it's not very significant. And then we have the channels. So let's see only the channels. If the image includes multiple channels, then you can activate or deactivate each channel to view the desired multicolor overlay for different structures. For this image, I only have 2 channels, the Alexa 488 and the Drag 5. So if I, for example, deactivate this channel, then I will see only the channel of Alexa. And if you need, you can also change the color. By default, it will give you some default colors, but in here you can also select a different color for the image. Okay. See the question in the chat, is not possible to see an intensity histogram. I think that is something we will see in the results once we run the analysis. Right now, this is only for visualization purposes of the image, but once we go to the image analysis, we will see the results that will be available. So we will see if that intensity histogram is what you are looking for, Beverly. Okay. All right, so same if we deactivate the Alexa channel and we have the Drag 5, so we will see only the nuclei in this case. Okay. For the channels, you can also use this slider to adjust the contrast of the image. We have minimum intensity and maximum intensity that you can use to adjust the channels. And we also have a slider to adjust the gamma value as well. But if you want, you could activate the auto contrast. So it will adjust by default the preferred contrast for a specific channel. Okay. All right, so that is about the image settings and the interface in general. Do we have any questions so far? All right, when setting image thresholds, is that global to the whole plate? Let's wait for the image analysis. Probably on there, I will talk about the global image in the settings for a global image when you run an analysis to the global, and also the batch analysis. If you're talking about the whole plate, to run the analysis in the whole plate, that will be for the batch analysis. Okay. All right. Any other questions? Okay. All right, actually, I see you have some questions. If you want to export the images for
Yeah, sorry.
publication.
It's all related to that first question. Like here
Okay.
we're adjusting the thresholds to see how the images look, right?
Mhm.
And then we can do sort of a mini montage sort of situation. But if we're like say doing these color thresholds in order to like include them in this SI of a paper or as part of a figure, then we usually try to have the same what's it called, intensity settings on every channel for all the images from the same plate.
Mhm. Okay.
And I'm asking if what you just showed us is this where we would do that, or do you have to do that in the analysis tab as you're alluding to, that we have to look at something in the analysis tab?
Probably in the analysis, because here in the channels controls, you are not changing, actually, as Beverly mentioned, you are not changing the data. So any settings that I do in here for the coloring, for the channel's intensity, when you log out and log in again, it will be restarted to the default settings in
Yeah,
the browse, right?
that's fine, and that's somewhat expected. It's just sort of like if we are just looking at we're looking at individual wells and we want to export the image for what's it called, but we don't want to manipulate the actual image or the data subsequent following up. We just want a presentation of uniform brightness, and auto contrast is on. Then we have to like butts with every single well, right? Or no?
Yes, but maybe I'm confused here if you're talking about the analysis results or the visualization of the image, right?
Visualization of the image for
Hmm,
to export.
okay. I don't think that's something you can do here because if I export right now, it won't export with the settings that you do here for the channels.
Okay.
We can give it a
I
try
think
because
this is
there's
just for like, this is just for when you're looking at the data on the screen. Like, it's not, this isn't the time when you would be making figures or,
Mhm.
that's a,
Yeah.
it's a different step. It's more the interface step where you're just like looking at things. So we're adjusting how you're looking at it, but it's not like for making a figure with data, you know?
Yeah. No. I
That's
mean
right.
that's fine. I'm just
Your
I
questions will be great, like at a different point, I think.
as I said, like I'm coming from MetaExpress, and in MetaExpress, the same place where you visualize the wells, the images of the wells in either montage or tiled format, or even like individually, is this
Mhm.
exact same place where you sort of set up the channel thresholds that you're going to
Mhm.
export, that you're going to like copy out and put into a publication.
Right. Okay.
It,
Yeah.
that's all. Is this
Yeah.
the same place where I do that, or and in that in MetaExpress you don't change the fundamental image or anything about it. You haven't touched the image file. You're just touched a copy of the image file, if
Mhm.
for lack of a better term.
Sure. Well, yes, this is the same place where you can change that, but if you're talking about export the image, then I'm not sure if that is the same place where you would do that.
I
Um,
mean, export the visualization of the image, right?
right,
Because
right.
your original image is all grayscale. Why would I be exporting a grayscale? I'm exporting this visualization with nice, pretty color, so I can include it.
Mhm. Okay. Let me give it some try with because in the simulator you can also export data. So I'm not sure here if the export will save the data with the specific visualization settings that you set in here. So I will run some tests about that actually, and I will follow up with you. Okay.
Cool.
Good.
I think Beverly and Matilda have succinctly put my question.
Okay. Yeah, how to save a copy of the image. Right, that is take into account the view settings. Yeah, yeah, I understand. Okay, I will take a note of that too. Just a moment. Okay. Any other questions? Okay, good. So let me go back to the presentation. All right. And now, well, here's just a reminder that we will use the ready-made solutions, but you already have that in your system. So in the ready-made solutions, we have some examples that you can use during the training. And also we have some other data that you can use to play around with the application after the training. You will notice that during this training, we will only use one measurement of all of this folder. So the idea of the rest of the data is that you have that on there, and you can get familiar with the application using that example data that I sent to you. Okay. All right, so now let's talk about some workflow icons. So we already saw the browse part, so where you can explore the images, you can see the results, you can see analysis sequences, screens, plates, measurements. Then we have these other two icons in here, workflow icons, that we will be using to run a specific analysis. The second one is the image analysis. So we are going to use this one to create and save analysis routines or sequences using building blocks. Then we have the batch analysis. This is to run analysis routines on entire plates or screens. So once you have the sequence from the image analysis part, we will apply that sequence to a plate or a screen using the batch analysis. Okay, a little bit about the data import and export of Signals Image Artist. In here, well, you can import images and data using the SEMA loader. It will allow you to upload from your local computer. Currently, those are the data types supported by SEMA, but I know that R&D is working on incorporating more formats for future releases. We don't know yet when exactly and what types they have on their roadmap. But I know they're working on making this list larger.
Quick question on clarification question is particularly. So when you say MDC store to TIFF or Metamorph TIFF, does this mean that we have to like say open MDC store tools to export the TIFF image even though we can find it originally in the file system? Do you follow?
No, please
So
the question again.
with our Image Express, we do our image acquisition. It gets saved to a local file system. I can navigate in Windows Explorer to that file location and look at the TIFFs. Is that the TIFFs that I can use to import into Signals, or do I need to go into MDC Store Tools, say export, and then have the export the exact same location where I could find it in Windows Explorer?
No, you can use the TIFF files that are locally in the computer that you saved. So same as we did for the sample data that we uploaded before. If you have a source folder where you have all of those TIFF images, you can just upload that folder, that local folder, using the SEMA loader. And just you will select in here what are those images that you want to upload, and then just click on upload. So you don't need to go to the external application and export from there. You can do that from the local file.
Okay, and it doesn't matter where in the file tree I select. Essentially, I could pick like the directory where like multiple plates of images have been acquired and are saved, and I can just select all the
Right.
individual experiments.
Yes. Yes.
Okay.
The best practice is that you put all of it in one folder, right? So you can just upload the folder with all of that data.
Okay.
Okay. All right. I see another question. What if you just have a TIFF images not especially for a software or database? For example, if I have data sets emailed to me from a user from a different microscope, they're just TIFF images, no metadata. Can I open them? Yes, that's right. Same as we did for the sample data. You can just select the folder where those TIFF images are, and that will be uploaded in the Signals Image Artist as a measurement. Okay. Any other question? All right, that's a great question, but really what data type do you put? Yeah, because we have some TIFFs in here. We would need to see if the data type of that specific images are supported. If that is not related to a specific software, then I'm not sure, to be honest. So let me take a note of that. You have very good questions. Okay. Any other questions? All right. Okay, so that is for the import, right? How you can transfer data from your local computer to Signals Image Artist. But we also have the export. The export is mostly for results after you run the analysis, but you can export any data, right? If I export the whole screen, then I will export the images, the results, the analysis sequence, any metadata that is associated to my my images. When I do the export, I can have well-level results or cell-level results. Well-level result is, for example, if you run your analysis and you decide that you want to save the results per well. So you will get only one file per plate, and you will get files for each analysis routine, but the results will be at the well level. Okay, so we'll get one file per plate with well level data. This will be different if we run an analysis and we decide to save the data per object or per cell. So that's why we call it cell level results. So in this case, you will get one file per well for each population instead of one file per plate. And then you will have results for every cell on that well. Okay. Any questions about that? Okay, good. Now let's see a little bit about the workflow to run image analysis. Probably you're already familiar with this analysis workflow, but we will just see this as an overview of the different steps. First, we need to input the image. So in here, we need to choose the image, but also we need to choose the channel and any visualization settings that we will use for our analysis. Then we have the image segmentation. So in here, we can identify nuclei, cytoplasm, or other sections or segments of my objects. Then we have the step to define regions of interest to do the analysis, right? So in here, once we have the nuclei identified or the cytoplasm, we can define if our region of interest is the cytoplasm, or if we have, for example, like a ring region that we want to use to run the analysis. Then we need to quantify the properties in those regions, properties like intensity, morphology, texture, and others. Once we quantify those properties in the regions of interest, we can calculate new properties from the previous results. So for example, if we have intensity, then we can use that intensity to calculate more properties based on those results. Then we have a step in here to identify subpopulations. So we can use filters based on properties, or we can use the phenologic, which is a machine learning algorithm that we can use to classify cells and identify those subpopulations. Okay, and then we have the calculate readout values. So here we can define what are those final results from the analysis that we want to capture in the results file. Okay, and then we will get those results that we will use to measure any biological effects depending on the purpose of the study that you're doing. Okay. Related to that, we have the building blocks. So we have all of these different steps that we will use in the software that we will call building blocks. The building blocks are elements which represent different analysis steps with a special or a specific function. So the idea here is to combine those different building blocks to build a complete image analysis that later we can apply in a batch analysis for other plates, other measurement, other data. Some of the types that we have of building blocks. First, we have some image segmentation building blocks. So we will use this to, well, for segmentation steps, which means to find the desired objects of your cells or the structures that you have in your images. Some examples are building blocks to find nuclei, find cytoplasm, cells, spots in other regions. Then we have feature quantification building blocks. So this is to quantify specific properties like intensity, morphology, texture, kinetic properties, cell painting, and we have some other properties in here. These building blocks will always start with the word calculate, right? So we'll get results for intensity, the shape, the morphology, the texture of your objects. We also have building blocks to create new channels. So we can calculate a new image from existing images, or we can apply filters. And we also have more building blocks in here, some additional properties to be calculated, like select cell region to define regions of interest, select population to filter objects according to their properties, modify population to identify subpopulations as well. And we will focus on this training mostly on the segmentation building blocks and the feature quantification building blocks. Probably we don't have the time to go over each building block that is in the software, but I will demo in here what we can do with some of them. And then with the rest of the building blocks, you can use the sample data or the CIMA help to read more about those and get more information about those building blocks. Okay, so this is the image analysis step by step that we will do in Signals Image Artist. So the first step will be to select a measurement in the data tree. So remember that the measurement is the folder that has the timestamp. Once we select the image, we can decide if we want to select one image or multiple fields, multiple planes. Um, the image that you select will be displayed in the image view section, and then you can start a new analysis. Then the next step is to add building blocks to define some parameters that we will call first level parameters, and also we have second level and third level parameters. So the first level parameters, you can use that to find objects, calculate properties, um, just to run the basics on specific building blocks. And then you can adjust second and third level parameters to optimize the results. Once you have the different building blocks, a recommendation in here is to toggle to multiple images and ensure that the sequence that you have, it applies to different treatments. Once you have that, you can define the final results or readout of the analysis. So what are those results that you want to record in the file of results once you run the analysis? And then you can always select a few other images to test and optimize the analysis once it is done. The final step in here will be to save the analysis. We have the option to save the analysis to the database in Signals Image Artist, or you can save the analysis as a local file in your computer. Okay. All right, we have a question in the chat. Can we get training tracking Phenologic? Can you view more than one image at the same time while building your analysis steps? For the first question, Phenologic will be covered on this training. So yes, we will cover that. Tracking is not covered on this training. So if you're interested on that, then my recommendation is to talk with Arturo or the project manager so they can give you more information about training for those topics. And the second question, yes, you can view more than one image at the same time. But at the same time will be if you're working with a global image of the same well of a plate. If you want to change to an image of another plate or another measurement, then you can do that, but it won't be at the same time, right? It's not like you will have both images at the same time in the image view. But you can change for those images as well to see how the analysis is applied to other images and other measurements or other plates. Okay. Any other questions? Okay. A few concepts before we go over the system. We will have some image analysis icons in here. We have icons to create a new analysis. We also have an icon to load an analysis. So if you have a local file with analysis sequence, that this is the format that we have in here, that AAS. If you have that file, you could load that into Signals Image Artist, and you can edit the sequence from there as well. Then we have the option to save the analysis. This icon is to save that as a local file, and the next icon is to save the analysis to the database. So it will be saved in Signals Image Artist under the current measurement. Okay. Then we have the show print version. So we have a printer-friendly format where you can see the display of all the building blocks sequence. So you can always click on this icon and you will have in another tab the different building block steps that you can download or print. And then the last icon is to run the analysis. So once you finish all the different building blocks, you can click on Run Analysis to perform the analysis on that specific image. But we will learn later in the training that you can also run a batch analysis if you want to run the analysis in a whole plate, for example. Okay. All right, when would you want to save the database versus local? I would say this local saving is mostly for users that have different image analysis tools. So if you have some other software for image analysis, you can just use the sequence in that format in different softwares. But if you're only using Signals Image Artist, then probably it's not that very useful to you. If for some reason you want to send that to someone else, right, then probably the save analysis as a local file is a good idea. But if not, the save analysis to database should work perfectly. You can run analysis from a analysis that is saved in the database without any problem. Okay. All right, this is the print version. So when you click on the show print version, you will see this page that will open in a new tab in your browser. It will have description of specific building blocks. So you will see that, for example, in this print version, we have find nuclei, find cytoplasm, calculate morphology properties, find spots, morphology properties, and intensity properties. So we can see the different steps in here as building blocks, and then we can see information about the input, the method we're using to calculate those properties, in the output. Okay. All right, then here how you can create more building blocks in the analysis. We have this plus icon to add more building blocks. So we will see that in the software in a moment. If you click on this plus icon, Image Artist will suggest the steps that you can start using. For example, when you have the input image, you will notice that the first building blocks that are suggested will be find nuclei, or find spots, or find cells, because you cannot calculate intensity, for example, if you have done any haven't done any segmentation steps like find nuclei, cytoplasm, or cells. So Signals Image Artist will always suggest a preferred order for those building blocks. You can choose others, but it will always suggest that to you when you click on this plus icon. Okay. And then some important concept in here is the progressive disclosure of the properties. When you create a new building block, we will have different levels of parameters. For example, this screenshot that we have in here is for morphology properties. all the different building blocks will have different parameters, but for this example, we have this first level where we can choose the population, the region, the method, the property prefix, some basics on how to run the building block as default. But if you need to adjust some other parameters, we have second level, third level, and sometimes fourth level parameters that you can use to optimize the results. So we'll change this to my arrow. So for example, if I click on this drop down that is in the method, it will display the second level parameters. So by default, all the different properties of this method to calculate morphology will be selected. But if I need, I can unselect some boxes in here if I don't need to calculate those properties. And if I click on this drop-down arrow, I will see third level parameters like profile, inner region, profile width. We have some filters like sliding parabola, text cursor that we can use for this specific building block. In this case, this building block has a fourth level parameter. So if I click on the sliding parabola, for example, or text cursor, I will have the sliding parabola parameters and the texture parameters that I can use for this building block. You see that Signals Image Artist by default will have some boxes checked and some other boxes that are not checked, but you can always display further level parameters to select or deselect some parameters that you don't want to calculate. Okay, so this will help you to dig deeper if needed. Right? I have another question here. Where can you get descriptions of all the level variables? Is it all in help? For example, what do they mean by actual and radial profile? Yes, definitely all of that is in the help. Here in the training, I will talk into more detail about the morphology properties, texture properties, so we can talk about those. But if in the future you need like a gradient description, that is also in the CIMA help. Okay. Another important concept in here is the tune parameters. So you will notice, and this will be easier to see it in software, so I will demo how to use this, just for you to know that we have something that is called the tune parameter that you can click on it, and you will have some, you will have like an option here in the middle of the image where you can have a preview of the different parameters or the different values without selecting those. So that will help you a lot to save some time. Okay. All right. I see that Beverly and Matilda are having some trouble hearing me. Is that the case of Axure S1? Okay. Seems it's fine again.
Yeah, we're fine.
Okay. Okay. Thank you. All right. So were those concepts that we just saw, let's begin with the different tasks. Before we begin, do you think it's a good moment to have a few minutes of break? I know it's been a lot of information probably, so we can take like 10 minutes and then we can take 10 minutes later on the training. Is that okay?
We don't need a break over here. What about you, Aksar and Aswad? Do you guys need a break?
Okay. All right, we're gonna skip the break. No, I mean, for me, it's okay. Don't worry, Aswad. If I need a break, I will let you know. It's just that sometimes I know it's a little hard to process a lot of information, but if at any point you need a break, please let me know, okay? Because I know 4 hours could be very long. Okay, so let's begin with the task number 1. In here, we're going to use this plate. You can see here in the presentation the name of the plate. It is called P013 CC cytoskeleton. That is part of the ready-made solutions that I shared with you. So you can search for that plate. First, we will find the image in here. We'll find the data and we will input the image. Before we start with the tasks and the example, I want to give you some background about this images, this data that we have in here that we will use for this example. We will use images of cells treated with different concentrations of thymicalcine. We will use different stains as channels to visualize different parts of the cells. And we will notice there are some differences depending on the concentration of the compound that we use. So the idea here is to notice those differences and build a sequence that can help us to analyze the shape, morphology, intensity of those specific cells before the concentration and after treating the cells with different concentration values. Okay. More context about this example. It has been studied that thymicalcine disturbs the equilibrium between microtubules assembly and disassembly because it sequesters alpha-beta tubulin dimers, components of the microtubules. So it results on increased number of mitotic cells, reduced cell number, and complete loss of cellular microtubules and MTOC, or microtubule organizing centers. So in here, we will analyze how the concentration of this compound will affect the cells, taking into consideration that it will affect the cytoskeleton of the cell. Okay, so we will use the first building block, which is the input image. This input image building block will come by default, and it is necessary to start the analysis. We will set up in here the initial parameters to start the analysis. Some of those parameters are the stack processing. So if you have multiple planes, for example, if you're using a 3D analysis image with multiple planes, you can use this stack processing to decide if you want to use those as individual planes or if you want to combine them for a 3D projection or 3D analysis. We also have the quick tune. The quick tune is to make the image analysis faster. So it does a crop of the image that you can use for testing to save computing time during the analysis. Sometimes, depending on the analysis that you're running, is not recommended because if you are doing a crop of the image, then that will in that case we will lose some of the structures and cells that are in our image. So it always depends on the purpose of your study and if you need to save some computing time during the analysis or not. We can also configure in here if we're using a global image. So if you have different fields in one well, you can build one global image using all the fields and you can run the analysis using that global image. Something important is that when you use a global image, the resolution of the image will be always affected because if you're combining all the images in one, the resolution will be affected and it could be that you will lose some of the details of the image. So again, it always depends. It's completely up to you on if you want to run the analysis as a global image and lose a little bit of resolution, or if you prefer to run the analysis for every field on a well and save the same resolution. When we create the global image, we also have the minimum global binding. So for this setting, we can also adjust the resolution of the global image. And the last step that we have in here is the comments. So in any building block, you can always add comments to the building block, and those comments will appear on the printed version if you want to print the steps of your building block or your sequence. Okay, so before we go to the task number 2, let's go back to Signals Image Artist. Okay, and we will start the analysis here. So again, the plate that we're using is the cytoskeleton. This one, the P013 CC cytoskeleton that I will highlight so if you can see it. Okay, and you can select the first measurement of that plate. Okay. If we navigate through the secondary navigation, we will see that the first well is the image without any concentration of the compound. And if we move at the right side, for example, we will see that the next wells, the cells will be a lot different, right? So we see that the cytoskeleton has been compromised because of the concentrations of the dimicolcine. Okay, so the idea here is that we will run the analysis to measure the morphology, the texture, intensity of the different wells. But to start the analysis, we will click on the first well, and then we need to click on this icon over here at the top that says image analysis. So right now we are in the browse part of the software. To start building the sequence of analysis, we need to go to the image analysis part in here. Okay, so we'll click on it. Okay, and immediately you will notice that we have the input image building block in here that it will always come by default when you start a new analysis sequence. Okay, in the input image, we have the channel group. So if you have different channels in different groups, you can choose in here the channel group. Same for the sequences. This is relevant for time series measurement with more than one sequence. Okay, in this case, we only have one sequence, but in here you can decide if you want to select the specific sequences. Then we have the flat field correction. So in here we can correct the image before the analysis. Same as we saw in the controls, we have none, basic, and advanced, depending on what is allowed in your image. Okay, so we can use basic, for example, or none, depending on what you prefer. Then we have the bright field correction. This will correct any bright channels, if you have any, to create a smooth visualization. If you don't have any bright channels, then you don't need to use this option. Then we have the stack processing. So if you have data or measurement with multiple planes, then you can use the stack processing to use those planes to calculate a maximum projection, and we can see like a 3D image, or if you want to perform a 3D analysis. Okay. Then we have the create global image. So if you select all the wells in here, for example, okay, and then I click on create global image, I will see in here that I have, in this case, I only have one. Well, this will work if you have multiple fields. In the case of, let me go back to input image. Okay, in the case if you have multiple wells, you can also use that, but here in the visualization, it will work if you have multiple fields. So for example, if I use this one, let me just change the data. Just a moment. Yeah, this one that has multiple fields. So if I select all fields and I have global image, I will have my global imaging here for all the fields if that is specific. Well, and that is what will be used during the analysis. Okay. Let me go back to the cytoskeleton. Okay. If you use the global image, we also have the global binding in here. So you can adjust the resolution of that global image from this parameter. Okay. Then we have the quick tune. As I mentioned before, the quick tune is if you want to have like a crop area of the image, so you can run the analysis only on that cropped area. That will help you to save some time, but you see sometimes it's not convenient because you will lose some details and objects that are part of the image. Okay, and then we have the comments. So if you need to enter any comments about this first building block that is for input image, you can write those in here, and that will display in the print version when you click on this icon over here that says show print version. Okay. Any questions about this? About the input image? Okay. All right, let's continue with the next building block. Oh, and Beverly, I don't know if that is what you were asking for, this histogram of intensity. You asked before. I cannot hear you. Yeah, I think it looks like what she was asking, but okay, let's continue with the next task. Okay, so next we have the task number 2. So now we will do an image segmentation. So once we have the input image, we can start with the image segmentation to detect nuclei, cytoplasm, cells, spots, and more regions in here. For this example, we will use the find nuclei and find cells. So we will use this building blocks to detect cell or nuclei according to your data. And same as we saw for the input image, we have first level parameters to refine that segmentation. So for example, in the find cells or find nuclei, we can decide which method we want to use to detect those structures. We can decide in here what is the output population name. By default, it will be cells or nuclei, but you can always change that depending on the data that you're using. And we also have secondary or second level parameters. So for example, if I display the method, I will be able to adjust a little bit the parameters of that method so we can optimize the results of that specific building block. We also have the find cytoplasm. So in here we can detect regions around the nuclei. So that will be the cell cytoplasm. Each cytoplasm is detected as a region on the image. It will have a higher intensity than the background. Same for the find nuclei and find cells. In find cytoplasm, we have the first level parameters where we can decide the channel, what is the nuclei population that we're using as reference to detect the cytoplasm, and the method. And then later we can adjust the secondary parameters for that specific method. Okay, so let's go back to Image Artist. Let's continue here with adding a new building block. So remember, to add a new building block, we need to click on this plus icon over here. Okay, it says add new building block after this position. The sorry, I think we have some echo. I will. All right, good. Thank you. Okay, so if we click on this building block icon, the software will suggest in here the next steps for my sequence. So you see that we only have available find nuclei, find spots, find cells, only the find building blocks because we need to do a segmentation step. And those that you have in bold are the most recommended building blocks that you need to use. So in this case, we will start with find nuclei. Okay. When we select find nuclei, we will have some first level parameters in here, like the channel, right? Automatically, Signals Image Artist will detect what is the channel that is used for nuclei. We have the region of interest population. Since this is the first segmentation step that we're doing, we don't have any population yet in here. And then we have the method. All right, so in this case, we have A, B, C, M. If you need more information about how this method is being applied to this building block, all of that information is in the SIMA help. Okay. And then in here, we can practice with the tune parameter. The tune parameter will work when you have a parameter that has this kind of hyperlink. When it is blue and it has a hyperlink, you can click on it. And here between the building block in the image, it will display the different values of the method in this case. And you can just easily, and I will zoom in so you can see the differences. You can easily hover over one of the methods and you will have a preview of how that will look on your image without selecting the method. So that will help you to save a lot of time because you can just hover over it. And once you're ready to select the one that you want, then you can just click on it and then click on the check mark. Okay, and it will be changed to method C. Okay. Then in here, if we display the secondary level parameters, then we can change the threshold to find nuclei and the area. Okay. We can keep displaying in here for third level parameters. We have splitting coefficient, individual threshold, contrast. And again, if you need information about any of these parameters now that works for the image in the final results, all of that information is in the SIMA help. Okay. All right, I see a question. To access the help for a specific topic, do you have to search through the whole help? Harmony lets you get help for specific things with, except, for example, find nuclei. Yeah, sure, it is separated into topics. So if I go to the SIMA help, for example, if we are talking about find nuclei, we can go to image analysis using building blocks. And then in here, you can navigate through find nuclei, actually. Let's see where it is here, building blocks, and then find nuclei, like that. Okay, or you can easily put here in the search find nuclei or the topic that you're looking for, and you can search for it. We also have an index that can help you to navigate through the help, and we have a glossary as well. Okay, let me go back to the interface. Okay. Then we have the output population name. By default will be nuclei, but for some reason you need to change the name for this output. You can always do that. This is free text, so it depends on how you want to call this population. And the comments. All right, you can always put comments on any building block. All right, that is for find nuclei. Now let's add another building block, but this time is to find cytoplasm. Okay, so again, we have the channel. ImageArtist will detect the channel that it is using to find the cytoplasm. We need a nuclei population for reference. Right now, we have what is called nuclei. If you put a different name, then you will have the name of the output in here. And then we have the method. That again, we can click on the word method, which is a hyperlink. Okay, and we can hover over a specific method and see the differences on my image and what better fits our analysis. Okay, so I can select this one, the method A, and click on the check mark. Okay. We can also do the same process for find cells, micronuclei, and any other objects in here. But for this example, let's just keep it with nuclei and cytoplasm. Do we have any questions about those two building blocks? Okay, another question, can you find cytoplasm if you don't have nuclei labeled? Is that what you use find cells for? For cytoplasm, you need the nuclei. You always need to find the nuclei. You can use the find cells, right? And the cells you can decide in region of interest population. If you don't want anyone in here, or if you don't have nuclei label, you can use none, but that will search for the cells. So if you're interested on the whole cell, then yes, you can use the find cells. But if you're interested on the cytoplasm only, then we need a nuclei. Okay. Something also important that I forgot to mention in here. Now that we have the building blocks for find nuclei, find cytoplasm, or find cells, you will notice that there is a new option in here in the image settings that says regions. So this can help you to visualize the regions as you prefer. So for example, right now we're looking at the cell and the border, but if we want to see the cytoplasm, for example, and that will be body, and we can select a color in here. Okay, we will see only the cytoplasm in purple, for example. Same for the nucleus, right? We can show here the nuclei, okay, with this color. That will be body. Okay, and we'll have something like this. Or probably we can change this to another color. Okay, so this will help you to visualize the different structures in a different way as you prefer. Okay, so let's get rid of this. Okay, and if I go to the find nuclei, automatically it will show me only the nuclei. If I go to the cytoplasm, it will show me the whole cell. It will be surrounded the cell border. Same for the cells, right? If I go to find cells, I will see the whole cells. All right, any other questions? Okay. Good, so let's continue with the task number 3. This is also part of the image segmentation. This time we will create a ring region surrounding the nuclei. Sometimes there might be subregions of interest for the analysis to study interactions that can happen in a specific cell region. So we will learn here that there is a building block that is called select cell region that we can use to create custom cell regions. So we already have nuclei, we have cytoplasm, but if for some reason you need to select like a region of interest for the rest of your analysis, you can do that using the select cell region. We have some different parameters that you need to define in here. So for the cell region, for example, we need to define the population that we're using as a reference for that region. The method could be different, and then we have some second level parameters depending on the method that you choose. And again, you can put an output region name depending on what you need. Okay. In the select cell region building block, we have two methods. We have the resize region with percentage or proportion, or the resize region with a constant value. If you use the proportion, then you can define like the percentage or the proportion between the width of the cytoplasm and the radius of the nucleus. Or you can use a constant value if you want like the ring region to be of a specific width. Okay, so let's go back to ImageArtist, and now we will add, and actually here I will remove the find cells because I only want to have find nuclei and find cytoplasm. And I will add a new building block in here for select cell region. Okay, this is the building block that we will use to define any custom regions of interest for our analysis. Okay. So first we have the population. Right now, the only population that we can use as a reference is the nuclei. If we have the find cells building block, then in here we will have cells or nuclei. Then we have the method. So if we want to use the proportion with the, which is a proportion between the width of the cytoplasm and the radius of the nucleus, or if we want to use the resize region with a constant value. So if we use the proportion, then we can decide in here the region type, if that will be cytoplasm region, membrane region, or a ring region, which is the one that we are going to use in this example. All right, so we'll select ring region. Okay. You will notice that here in the image, we have the default ring regions. Then I can adjust the outer border and inner border. Again, since this is a hyperlink, I can click on it and I can use the tune parameter to just hover over the different values, and I will have some kind of preview of the different values, right? So I can see how the outer border will modify my ring region. Okay, so we can use this one, for example, for outer border. And then we can repeat the same process for inner border. Okay, so we have 40 as default, but we can see the difference if we adjust the inner border of my nuclei. Sorry, of my ring region. Okay. We can also change the output name. By default, it will be called ring region, but if you have a specific name for this region, you can put that name in here. Okay. Any questions about this? Okay. Let's go back to the presentation.
Actually, we do have a question over here.
Yeah. Okay.
I am using something similar in a cell painting module in Harmony. It's also using the different regions of the cell.
Mhm.
I am guessing it will be similar. Cell painting module will be similar here, yeah. In Signal ImageArtist is using like preset image regions. The cell painting module here in Signals.
Yes, we have a building block for cell painting that is not part of this training, but you can explore more about the cell painting building block, and you can see the parameters that you can adjust on there for the region and some other parameters.
Just one question there. For this cell painting module, I also need to find nucleus first, yes?
Yeah, that's right.
But do I also need to find cytoplasm or any other, or a nucleus is sufficient to run the cell painting properties? Or I should also find the other?
Yeah, it always depends on the purpose of your study, right? Or what you need, but you need at least one segmentation building block, right? Like fine nuclei or fine cells in this case. But it depends on what.
Yeah. I understand. But it will. Okay. I need just one. Find
Right.
nucleus or find cytoplasm. Okay.
Yeah.
Cell.
You,
Yeah.
you
Okay.
see that when we have the input image, we can only select find or segmentation building blocks, right? So you cannot start any other analysis like cell painting or any other analysis if you don't have at least one segmentation step. So, yeah.
Thank you.
Sure. Okay, any other question? Okay. All right, let's continue here with the presentation. Then we have the task number 4. Here we will calculate intensity properties. So we will start with that quantification building blocks that will give us numeric values or numeric results about intensity in this case. But we will also continue with morphology, with texture, and some other parameters that we can, or properties that we can calculate using the different building blocks. Okay, so for this building block, the purpose is to calculate intensity of a region of interest. So that's why it's very important to always define the segmentation, and also if you have a specific region of interest, like the ring region that we created just a moment ago, it's important that you do all of those steps before you actually calculate the intensity. Here we have again some different parameter levels to define the channel, the method, population, the region of interest, and that will help us to calculate different numeric properties associated to the intensity of this region, right? So here, depending on the method, in the second level parameters, we can decide if we want the mean, the standard deviation, coefficient of variance, or any other properties to be calculated in here. Okay. So this will generate, or all of those numeric values will be considered cell-based data. Examples of some things that signals image artists can calculate with intensity is the mean, standard deviation, coefficient of variance, median, quantile. Oh, sorry. And we also have some in here, maximum and minimum. What the building block will do here is it will calculate the minimum bright pixel of your objects, of the different cells, and the maximum bright pixel of that cell. So with that information, it will calculate the intensity mean and the rest of the different parameters in here. Okay. So let's go back to Signals Image Artist. And now in the Select Cell Region building block, we will click again on this plus icon that says Add New Building Block after this position. Okay, and we will have in here Calculate Intensity Properties. This is the one that we will be using. Okay, some of the first parameters, we have the channel. So depending on the region of interest, if you are calculating intensity in the nuclei, then probably you need to change the channel in here. Since we are using the ring region that is in the cytoplasm, I will use the Alexa channel. Population, this will be like the reference that the system will use to count the number of objects. So nuclei will be used to calculate how many objects you have in there. But then we need to define the region. So where do we actually want to calculate that intensity? Is that in the nucleus, the whole cell, the membrane, the cytoplasm, or the ring region that we created just a moment ago? Okay, so in this case, I will click on ring region because that is the region of interest that I want to use to calculate any intensity. Okay. Then we have the method. For intensity, right now we only have one method, which is the standard, so we don't need to change anything in here. But if we click on this drop-down arrow, we will display second level parameters and third level parameters. So by default, it will calculate the mean, but if I want, I can calculate the standard deviation, coefficient of variance, median, sum, maximum, and minimum. Okay, so let's do something like this. Okay, then we have the property prefix. It will be called intensity ring region and then the name of the channel. But again, you can always change the name of the prefix for this property. And then if we see the table view, we will see the results in here. And if we go to properties nuclei, we will see the different values of mean, standard deviation, maximum, minimum, and sum for every object. All right. And if I click on one of these objects, let's click on another one. Okay, you will notice here in the image that it will be highlighted. So this object number 6 is this cell over here. Here at the right side, we have the overlays that you can also use to change the way you view highlighted objects. Right now we have a solid with orange, but if you want, you can always change the way you see highlighted objects. Okay. All right, so that's for intensity. Do we have any questions about that? Okay. Good. All right. Okay, the next part, the second part of this training is the powerful image analysis. So here we will see some other building blocks and tools that we can use to run some advanced image analysis. Um,
Hey, do you
Yep.
think now will be a good time to take a small break?
That's what, yeah, what
Yeah.
I was going to say.
Okay.
I will do a pause right now. I think it's important we have that break so we don't lose any attention, and also I need to take some water. So is I think 15, we don't have a lot to cover right now, so 15 minutes, is that okay? Or do you think you need less than that? All right. Okay, so I will put a timer here on the screen so all of us can see the timer, and we will be back after, yeah, let's do 10 minutes so we don't run without any time at the end of the training. So I will put a timer of 10 minutes here on the screen. And as soon as it reaches 0, we'll be back to cover the second part of the training. Okay, time's up, so we're back. Can I get confirmation that all of you are back before we start? Maybe in the chat or with the thumbs up.
I
Okay,
can report that I'm here. Matilda had to leave. I saw.
okay, no problem. That's what we have the recording for. Okay, good. So let's continue with the powerful image analysis tools. So here we are going to cover these three advanced properties or advanced building blocks. First, we have the texture analysis. So we will use this to identify the texture of a region of interest using quantification of intensity patterns. We also have the advanced star morphology analysis. This will give us an idea of, or will give us a sorry, my microphone was muted twice, but can you hear me now? Yes. Okay, good. Thank you. All right, so the second one. Sorry, I don't know what's going on that it keeps muting me. Let me try again, just a moment. Okay. All right, so the advanced star morphology will give us a description of any cellular phenotypes based on fluorescence signal. And then we have the phenologic. The phenologic, it will help on the segmentation and classification of cells using machine learning algorithm. Okay, so we'll see more detail, the three of them. And we will start first with the advanced texture properties. So first, let's talk about what is texture. Probably we'll know what a texture is, but technically a texture is the appearance or consistency of a surface. So the first image has a more irregular texture than the second one. And we can say this just by looking at the image because of the intensity patterns. So the first image will have different intensities that make us think that texture is irregular. And the second intensity pattern will give us an idea of a smooth texture. In the case of a cell, the texture analysis can give us an idea of how the fibers and surfaces of structures will look like. The texture analysis will include calculations like intensity, morphology, and the measurement of patterns to define the texture. This will be very useful to identify any kind of phenotypic changes. For example, the example that we're using right now for the cytoskeleton, with this kind of building block, we can measure or we can identify those phenotypic changes depending on the concentration of compound or a specific compound that we are using in a cell. The method that we'll use to evaluate texture, it is called SER or SER. This method will use intensity pattern that is analyzed using spots and holes. So we can see here in the image that a hole will be like a pixel with low intensity surrounded by high intensity pixels. And the spot will be a pixel with high intensity surrounded by low intensity pixels. So this method will use mathematical modeling to fit the texture to this spot equation. So based on this hole and spot patterns, the model will generate different intensity patterns to describe the texture of a cellular surface. So besides of spot and hole, we will have ridge, valley, saddle, edge, bright or dark, depending on how intense the pixels are in a specific object and how those different pixels are arranged according to that spot and hole logic. Okay. In this building block, we can also adjust or tune the scale to fit the pattern to our image and get on that way we can get better results. So Signals Image Artist will always suggest a value for this type of scales, but if for some reason the scale needs to be adjusted because of the protocol, then you can do that from here, and that will change how a specific pattern will be applied to the image. Okay, so with that explanation, we can go through the task number 5, which is to calculate texture properties. So in here, we will calculate the texture properties of the cytoplasm. With this building block, we can visualize the different patterns against the original image, as you can see here. So we have the original image, and then we have all the different filtered images using filters for spot, hole, edge, ridge, and so on. So we can decide in here, just by looking at the images, which pattern better fits our structures. We will also get some numeric values. So with those numeric values, we can also make decisions about which pattern better describes the texture of our cells. Okay. So just a comment about this method, it will help us to decide which texture better describes a structure. So which pattern will better describe a filament or any kind of a structure that is part of our objects. Okay, so let's go back to Signals Image Artist. Okay, and let's add the next building block in here by clicking on the plus icon for calculate texture properties, right? This is the one that we will use in here. Okay, it will be added in here. We have, again, the channel, we have the population, and we have the region of interest. If we want to calculate the texture in the whole cell, then in here we choose cell. If our interest is to have the texture of the membrane, we can choose that in here, or the cytoplasm. And actually for this example, we will use the cytoplasm. Okay. Then in the method, we can select 3 different methods. In this training, we will only focus on the SERP features, but in the Sima Help, we can read more about the other methods that we have to calculate texture. And if we click on this dropdown, we will display the second level parameters and the third level parameters, so we can change the scale, we can change the normalization, and also we can select in here which filters we want to apply to our image. In the image view, we can also select the different tabs that we have here at the top to see or have a better visualization of how a filter is applied to the original image. So if I click on Zero Spot, for example, okay, I will see here it probably doesn't really fits our image, and we can keep going with Zero Hole, Zero Edge, right? We see that edge, it looks good, right? It's describing, it's better describing the texture of my cytoplasm. We have ridge, valley, saddle, bright, and dark. Okay. We can always change the scale, right? For some reason we changed the scale in here. Probably we will see some different results. So for example, if I put here 2, we will see some differences in the images, the filtered images. Okay. All right, I will go back to the default, so I will have 0. Then in the summary, here in the table view, I will display here the table view. If we go to the tab of nuclei, we will see all of those values for morphology, for the search features of morphology. Sorry, for texture. So for example, in here we have spot, we have hole, edge, ridge for every object. If we go to the summary, then we can see here the values for all the objects as a mean. So here we can see, and usually the highest value here will be the feature that better describes our texture. So we can easily see that the edge is the feature that right now is better describing the texture of our cytoplasm. Okay, and we can also see that by looking at the image, the filtered image that is describing the texture. Okay, I see a question in the chat. I have run some analysis where the texture didn't visually make sense to me, but the quantification ended up being really interesting. What do you think of the situations? In my case, a particular texture was predictive of a particular treatment. Okay, even though it didn't make logical sense upfront, why to measure that particular texture? I had just measured everything. Also, can you explain about the kernel normalization? Okay, about the visualization and the quantification, I would say that uh, it also depends on the scale that you are using and also, well, the normalization, right? For the normalization, if you want like a better description of what the kernel normalization will do or the difference between region intensity or unnormalized, then my recommendation is to read about that in the help guide. The CIMA help. We that will also give you, sometimes depending on the parameters, will give you equations, formulas that the software is using to calculate this kind of parameters in this case. So I don't have like a lot of experience in analysis of images, so I don't know if when that doesn't make sense, if the quantification doesn't make sense with the visualization, but I would say that you will always need to adjust the scale and the normalization, and it also depends on the purpose of the study. If the numbers are very important for the purpose of your study, then probably the quantification that you have in here is enough. But if depending on the study that you're running, if the visualization is more important, then we need to play with the scale and normalization to have a better tooling here. Okay. Sorry, I cannot give like a lot of description about the kernel normalization, but that information is in the CIMA help, and I can also make note of that so I can do a follow up with you, Beverly. So kernel normalization. Okay. Any other questions? Okay. All right. So we don't have any questions about the texture. Let's continue here with the next task. Okay, so next we have the morphology. In the morphology, we can have an idea of the shape and size of a specific object. Here we could calculate morphology for nuclei, cytoplasm, the whole cell, or specific regions of interest. In the morphology, we have two different methods that we will cover on this training. We have the standard method and we have the advanced STAR method. The standard method to calculate morphology will include properties like area, roundness, perimeter, length, ratio, width as well. Right? So all of these properties are basic properties, part of the standard method that can give us an idea of the size, mostly the size, and the shape of our cells. Okay, so let's do the example with the standard first before we go to the advanced method. So we'll click on this plus icon for texture properties. And then we click on calculate morphology properties. Okay. First, we have the population, then we have the region. So we can decide in here which is the region of interest that we want to use to have a better description of the morphology. In this case, we can use the whole cell, right? Because we want to measure what is the morphology of the whole cell. And then we have the method. So as you can see, we have a standard and we have the star. The standard is the first one that we will do. So in here, we will include properties like area, roundness, perimeter, width, length, and ratio width to length. Okay, and all of those values will be displayed here in the table view. Okay. And if we want to see the area, roundness, and all of those properties for a specific object, then we can always go to the properties nuclei. And in here we can have the object number, and we can see the value for area, roundness, perimeter, and so on. Okay, that is for the standard method. Now let's see the difference with the star method. Okay, so the advanced star morphology method, it will calculate morphology properties for phenotype classification, same as the other methods. The morphology in the star method includes the outer shape of the objects and also the distribution of intensity inside of the objects. So for example, with the star, we can calculate properties like symmetry thanks to the distribution of the intensity inside of the objects. We have different parameter levels in here. So we saw this before. We have the first level where we choose the population, region, the method. The second level is choosing here the specific properties that we want to calculate. Third level is tuning for profile, and we will talk about that in more detail in just a moment. And the fourth level parameter will be for sliding parabola and texture, which are some filters that we can use to optimize the results of our built-in block. So let's talk a little bit about each of the properties that the start method will calculate. The first one is the symmetry. Intensity is used for this property. It measures 8 properties reflecting the symmetry of the intensity distribution inside of each object. So you can see the 8 properties here in this table. You see that the properties will have number 0 or 1 and then number 2, 3, 4, or 5. Okay, that is because the 0 or 1 will represent the intensity decay in radial direction, all right? So distribution of the intensity. And the numbers from 2 to 5 will describe the number of repetitions in circular direction. Okay, so we can have a symmetry with 2 repetitions or like a more triangular symmetry with 3 repetitions and so on. So depending on the intensity decay in radial direction and the circular repetitions, we can combine this to have 8 possible properties that will describe the symmetry of our object. Okay. Then we have the threshold compactness. Compactness is calculated to describe if an object is fragmented into many parts, applying a threshold that indicates the bright regions to be considered. So for example, if we have threshold compactness of 40%, it will indicate that only regions with more than 40% of the maximum brightness are considered. And if we have low compactness, then that means that the region is fragmented into many parts with some irregular borders. If it has high compactness, then you will see that as one object, almost as one object. Okay. Image artists will use 4 different percentages to describe the object compactness. So we have 30%, 40%, 50%, and 60%, depending on the regions, the percentage of regions with maximum brightness to be considered. Oh, something important forgot here. The threshold compactness is described using this formula. So this is an example of some things that you will find in the CIMA help. If you need more detailed information about the formula that is used and the method that is using, and how those numeric values will be represented in the results, all of that information is in the CIMA help. Okay, then we have the axial properties. So to understand the axial properties in the STAR method, we need to compare in here and see the difference between the classical morphology properties and the STAR axial properties. So we can see that the classical morphology properties will calculate the width and length of the object like this. But the STAR method, in this case, will look to fit the largest possible ellipse into the object and quantify the length and length ratio of the two principal axes of the object. So in that way, you will obtain the axial small length and the axial length ratio, which is the ration of the short hair to the longer axis lengths. Okay, so this will give you like better fit of the size of your object because of this of the method that we're using, which is an advanced method. Okay, then we have the radial properties. These are properties that characterize the intensity distribution in radial direction. So for this property, we will get some values in here. We can get the radial mean, which is the mean radius of the object based on the intensity. The radial relative deviation, so this will describe if the fluorescence distribution is homogeneous. And the radial mean ratio, which is the ration between the radial mean of the filtered image and the original image. Because in the morphology method, you can also use filtered images and compare those to the original image. So if you do that, you can have this radial mean ratio to compare the radial mean of the original image and the radial mean of the filtered image. Okay. All right, then we have the profile. The profile will measure the fluorescence intensity using like predefined regions represented with profile images. So in here, we will have 5 possible profiles. And we can see that here in the image. So we have the profile 1, profile 2, 3, 4, and 5. The profile 1 will be like the inner side of the membrane. The profile number 2 will be the inside of the cytoplasm. The number 3 is the outside of the nuclear membrane. The number 4 is the inner side of the nuclear membrane. And then the last profile will be the inside of the nucleus. Okay. In this building block, we can also decide if we want to use a profile inner region or not. If we select a profile inner region that, for example, in a cell, the profile inner region will be the nucleus. So if we do that, then the application will calculate these 5 profiles, right? Considering the nucleus and that we are calculating profile 1 and 2 for the membrane, and 3, 4, and 5 for the nucleus. But if you don't choose an inner region, then the system will calculate only 2 profiles. So that will be the inner side of the membrane and just the inside of the cell, right? Without considering the nuclear membrane. And we will see that in the software, so you can see that more clear. Okay. Something also important in the star morphology method is that you can use filtered images. The star properties in this case can be combined with 2 filters. We have the sliding parabola and the Sir texture filters. The sliding parabola is a background subtraction algorithm that we can apply just to correct the background of the image. And the Sir texture filters will be the same Sir patterns that we saw a moment ago for the texture building block. So you can combine these filters with the morphology star method. And the main benefit of using that is that it will increase the obtained information, and it will also allow you to study where a specific texture is located inside of the cell. Okay. So about the sliding parabola, this is the background subtraction algorithm that we can use to correct the background of the image. We can change the size of the curvature, and that will change how the background will be subtracted in here. And then we will get the filter or the raw data in the filtered image with the sliding parabola, just with the data that is of our interest. Okay. In the sliding parabola, we also have this checkbox in here that is called Use for Center. This option will determine if the original or the sliding parabola filtered image is used when calculating the mass center of an object. So if we do that, it will affect the properties like symmetry, axial, and radial properties. Okay, I see a couple of questions in the chat, so I will read those. When we calculate morphology properties, is it recursive on the regions of interest? Like here, we've set the population to be the cells, but can we add the module again for, say, a nuclear population, or does the cell population include the nuclear mask? So we have 2 different concepts in here, right? The population and the region of interest. The population will define mostly the identification of the objects. So if you use the nuclei as the population and you have 150 nuclei in your image, then it will calculate 150 values for the texture, right? Because it's using the nuclei as your population, as the number of the final objects. Now, the region of interest is that region that the software will use to calculate the morphology. So if you want to have morphology data of the whole cell, then you can use the cell as the region of interest, and that will include the nuclear mask as well. If you just want to calculate morphology in the cytoplasm, then you use the cytoplasm as the region of interest. Okay. And about the second question, that does the cell population include the nuclear mask? I will say yes, but I will change that population for cell region of interest, right? So that cell region of interest will include the nuclei. Hope that answers the question. If not, I can also show that in the software. Just a moment. We have another question. How does the star profile compare to defining our own subregions and calculated intensities in there? Beryl, can you unmute yourself and elaborate a little bit about that one? What do you mean about defining your own subregions?
Yeah, sure. So, when you were telling about the profile. It's saying, okay, here's just inside the membrane, here's the middle of the cytoplasm, here's just outside the nucleus, just inside the nucleus, and then the middle of the nucleus. That's very similar to earlier when we were defining like the ring region, for example. Basically, the ring region we made earlier was kind of the sum of the just outside the nuclear membrane and just inside the nuclear membrane. So it's almost like two different ways to get at the same answer. You know, like I doubt you need to do both, like define all those ring regions and then do the profiles of all those ring regions. You know, it's it seems like they're measuring like a similar thing. So I just wonder, do you have any comments on when would you want to use one or the other, or are there some situations where one performs better than another? Is one faster or slower? You know, does one take more manual optimizing? Like how do these things compare? I don't know if you have any information on that.
Right. Okay. Same. Yeah, I understand. In that case, the profile is, I wouldn't say it's the same, because the profile will give you like a numeric value of things that the system is calculated, and actually we'll see that in the software in just a moment. So you can see the difference. If your interest is to calculate morphology, and let's say morphology, I'm referring to symmetry, axial properties, radial properties. If you want all of those values per subregion, then my recommendation is to use the segmentation steps that we saw before with the building blocks, and then you just calculate morphology properties for each subregion. Because if you use the profiles, it will give you a numeric value or a filtered image. But you will see in just a moment that it won't give you like detailed information about each subregion. So actually, I have a few other slides, and I will show that in the software so you can see the difference. Okay. Any other questions? Okay. Good. All right. So yeah, actually, let's go now to ImageArtist. Okay. Let's do the morphology properties. We already have the building block in here. Remember that we did it with the standard method. Now this time I will just override this building block, and I will choose the start method instead. Okay. So this is the comment about the difference between population and region that I said before. You see that population right now, I only have nuclei because that is the population that I'm using to identify my number of objects in the image. But then I have the region. The region is different because in here I will decide which region of the cell I want to use to calculate those morphology properties. If that will be only in the nucleus, the whole cell, which includes the nucleus, membrane, cytoplasm, or the ring region that we defined before. So actually, let's do it on the whole cell. Okay. And then we can see here the different properties for the method. Again, we have the channel, and then we have properties like symmetry, threshold compactness, axial, radial, and profile properties. If I dig deeper in here, I can decide if I want to use a profile inner region for the nucleus, or if I don't want that, I can just uncheck the box, and the system will give me only two possible profiles. And we can explore the profiles here at the top of the image. We have these tabs where we can see the profile number 1, which is the inner side of the membrane, and the profile number 2, which is the inside of the object. If we define the profile inner region for nucleus, then we will have inner side of the membrane, inside of the cytoplasm, outside of the nuclear membrane, inner side of the nuclear membrane, and inside of the nucleus. Okay, so it depends on if you choose that inner region. And then in here we have this light and parabola and the texture. So these are filters that you can apply if you want to correct the background. Now we have another tab in here for light and parabola that we can use to compare the original image, which is this one, with the slide and parabola filtered image with that background subtraction algorithm. Okay, and the same for the texture. We can combine the texture properties with morphology, so we can have more information about where a specific texture is located. So we can use the edge, for example, and we will have that calculated in here as well. All right, so now let's take a look here in the properties that we are calculating. Okay. So if I scroll down, I will start here with symmetry. All right, so we have all the different symmetry properties in here. Remember that we are combining 0 and 1 for the intensity decay in radial direction, and from 2 to 5 for the number of repetitions. Okay, then we have threshold compactness for the different percentage values, axial small length and length ratio, radial mean, radial relative deviation, and then we have the profiles, right? So you can see here, Beverly, that profiles are a numeric value that right now I don't recall exactly what this value represents. That is in the CIMA help, but you will see that if you need, for example, the symmetry of the membrane and the symmetry of the nucleus, that's something you won't get with the profiles. You will need to have the subregion for membrane and a region for nuclei and calculate those properties separately if you want to have both on there. Okay. Then in here we have the filters. So this is the slide and parabola filter. If it says SP, then this is the slide and parabola. So we can compare symmetry values of slide and parabola filtered image against the symmetry values of the original image. Okay, and the same for the rest of the properties, right? This is all for slide and parabola filters, and then we have the same for the edge filter. Okay, so it's calculating morphology now using that filter of the edge pattern for the zero texture method. Okay. All right, do we have any questions about that? Beverly, you have any follow-up questions about the profile? I know that I don't have the exact description of this value, but we can review that in the CIMA help as well.
Yeah, I think I'm fine. It'll just take some playing with to learn all about it. All right, any other questions? Good. Okay, so let's continue here. Oh, I think I saw another question. Oh no, that was the last one. Okay. Then we have some guidelines here that we need to take into consideration when we use the START method. For example, if you are wondering when to use it, some recommendations that we have in here is that to use the START morphology method when the location of the texture areas inside of an object are important to identify as a biological effect. Also, in any case where the shape and structure of objects should be used to classify them. So if you later are interested on classifying cells based on the texture, or sorry, based on the morphology like symmetry, size, then you can use the STAR morphology for that. And also when the intensity measurements of specific fluorescent markers in different regions are not sufficient to measure the biological effect of interest. Now, how to use the method. Always select all the property classes since rational selection is difficult. So before you actually get the numeric results, it could be difficult to decide if you want to calculate symmetry, axial properties, radial properties. So the best practice in here is to always select all the different properties. Then you can see the numeric values and make decisions based on that.
Another recommendation is to use texture filters if you observe changes in cellular texture in different treatment conditions. So if that is of your interest according to your study, then that combination between the texture filters could be very useful. Now here we have a recommended feature set, because if you calculate all the morphology properties with the slider parabola and with all the SIR features, you will get 219 results. So if you want to have less properties and start from less than that, then our recommendation is to use all the star properties plus the slider parabola plus spot and ridge. That will give you 87 calculated properties instead of the 219 properties, and then you can go from there and make decisions about what other filters you can add or delete depending on the data that you have. Okay. Another recommendation in here is to use the STAR method with the phenologic, so you can classify cells according to their morphology as well. Okay. Any questions so far about the STAR morphology method? Okay. So let's continue with the task number 7, which is select population. So here we will use the phenologic algorithm to classify objects. Phenologic is a supervised training machine learning algorithm that can be trained to discriminate cellular phenotypes. So supervised training means that you will need some input from you so you can train the system, and then the algorithm will apply those training points to other data sets. So for example, we need to choose in here which is class A and class B according to our rational input. Then phenologic will generate this plot to separate class A from class B. And then if we apply that algorithm to another plate or another measurement, phenologic will give us this result where it will have the class A and class B according to the training that you did for the system. Okay, it contains 2 packages. We have the smart classification and the image segmentation. So the image segmentation will be used in a building block that is called find texture regions, where we can teach the software to recognize different cell populations or different regions based on texture. So this example that we have in here is a background correction example that uses the building block find texture region with phenologic using the method that is called split into classes. We can see the method here. So one class is the cell population, so all the different cells, and the second class is the background, right? So that's an example. The second part of the phenologic is the classification or the smart classification package. This is used with the select population building block for the method that is called linear classifier. So here we will teach the software how to recognize different cells automatically based on the training that we do for them. We can also combine the phenologic with properties. So it's not only about the rational input that you do, it's also about the properties that we previously calculated for intensity, morphology, texture. So the system will identify those significant properties that are used for classification. Okay, it will calculate a linear combination of the properties So values less than 0 will be class A. We'll use this formula here, and values less than 0 will be class A, and values higher than 0 will be class B. You will see a plot like this that is generated that will be used to distribute um, both classes. And also a new property will appear to compare the class A versus the class B. It is called the regression AB. And also the phenologic will identify relevant properties after the classification. So after we run the supervised training, it will identify those properties that are significant, that will help the system to classify cells depending on their properties. Okay. I see a question in the chat. Can you confirm it's only using the properties you have already calculated in the steps above? So for example, if you never instructed to measure the textures, then the classifier would use texture info. That's right. Okay. Now, the phenologic will use not only the properties, but also your input, right? So if, in this case, texture probably is not a good example, but if you have some other parameters that you use to classify the cells, that you visually see those different, and you want to generate some kind of input that this A is different from this B, then Phenologic will capture those training points that we will see in just a moment. But if we are talking about more advanced properties like the texture, then yes, first you need to have a building block for texture so they can it can use it for that. Okay. Here we have some guidelines. In general, Phenologic will need reasonable inputs from the user to make a clear separation between objects. So you can modify, always you can modify the training and add more properties, or change the training point so you can get the expected results. Something important here is to be very thorough with the training. So it's important to take some time, invest time in the training so the system can have very reasonable input when you do the classification. Okay. This is an example of an object of class A and class B and how it will look like according to our selection. And we could also find some borderline objects in the middle of the plot. And sometimes the system will need user input to correct those results. It's not that you can manually correct those results, but it's always a matter of changing a little bit the training points, changing the properties, so you can get better results for borderline cases. Now, some guidelines about how to set the training points. Here, the recommendation is to select objects that clearly belong to a certain class. So do not select borderline cases because that will generate confusion to the machine learning algorithm. Also, select objects from different wells, from different fields and treatments, so you can have more cells and objects to choose from. Also, study your populations in the scatter plot that will be generated. And the recommendation in here is to use more than 100 training points per class. So that's why we say that we need to invest time to train the system, because 100 training points per class could be a lot, but the good thing in here is that that training will be later used for other objects, other plates. So it's just something that you can do just once. Another recommendation is to clean up the analysis sequences, remove any properties that are not used for classification or that you don't think are useful for the classification. That will remove all kind of noise in the classification when Phenologic runs the algorithm. Okay. All right, I have another question here. Can you comment on oversampling if your train set is too large? I've heard that it's also bad. In addition to it, it's bad if the training set is too small, the more obvious case. I would say the training set, if it's too large, you need to analyze if maybe you are choosing some borderline objects, because I think that's why will generate some kind of oversampling errors, because if you have cases that clearly belongs to class A and some cases that clearly belong to class B, if you start doing a lot of training points, probably you will fall into choosing borderline cases. So I would say that's my comment about doing a lot of training points. Um, Obviously, training set too small is bad because the algorithm won't get a lot of information to work with. So that's why we always use the number of 100 training points per class. It could be more than that, but that will be like the recommendation that we have for the software. Okay. Any other questions before we go over the system? Okay. All right, so now we will add a new building block in here for select population. Okay. Now this select population building block is not only for the phenologic. We will do the example with phenologic and how you can perform the training set. But in here, we also have some other methods. For example, we have filter by properties. So if you want to classify using only properties, but not the phenologic, then you can use this method and select in here the different properties and the value that is used as a limit to do the classification. Okay, so that's one option. Let's do the example here with the linear classifier, which is the phenologic algorithm method. Okay. As soon as you click on the linear classifier, you will see here in the middle of the building block and the image, the training set or training radio button here that you can use to define the class A and the class B. You can also define the number of classes. We have a maximum of 6 classes that you can do with phenologic. For this example, we will do only 2 of them. And if we display the second level parameter in here with this drop down, oh, that's right, I need to get rid of the training so I can show you that. Okay, no problem. We will do the training and then in a moment I will show you the different properties in here that we can choose from. So for the training, we need reasonable input in here. Probably for this example, I wouldn't do any reasonable input, but let's see that class A will be like this. Cells over here. Okay, we can move around the image and get some other cells. Remember that the recommendation is to have 100 training points per class. So probably you will need to select other wells, other fields if that's the case. So you can select training points from there as well. Okay. Once you finish the training for class A, then we can go to class B. So we can select the radio button for class B and we can start the training from there. Okay, so class B, this is class B, class B. Okay, something like that. Once we finish the training points, then we just click on this check mark that says accept and exit the training. Okay, see if it seems like I missed the class A. Let's do it again. All right. So now the rest of the cells will be classified as class B or class A, depending on the input that we did for the algorithm. And here we'll have a tab for class A and another tab for class B. If we need to go back and correct some training points, then we can just click again on train segmentation routine. Okay. And here we will have the scatter plot. If we click on A versus V, then we'll have the scatter plot to see all the different objects of class A and class B. And also now in here, we can display the properties. And this is what we talked about before about cleaning the sequence. So if you think there are some properties in here that are not useful for the classification, then you can uncheck the boxes for those specific properties. Okay. And here in the A versus V in the table view, we can also see information about the properties that are relevant, that the system has identified as relevant according to the training set that you just did. Okay. All right, I see another question. Is there a way to select cells for training using select population filter by properties? For unbiased automated training set selection, like if I want to pick cells based on some really obvious features, but use more subtle features in the learn algorithm. No, if you use the filter by property, you will only use the property values, and that's it. If you need to select the specific cells for training purposes, then we need to use the linear classifier instead. I recommend you to read more about the other methods in here. I'm not familiar with the common filters and the select by mask. So I recommend you to read about those in the CIMA help to see if maybe that better fits what you are looking for. Okay, another questions. What if you want to add some properties and then redo the classifier, but you still want to pick the same cells for the training set? Will it forget what cells were selected? No. If I go back to the training, okay, and if I want to add more properties, or oh, sorry, Beverly, probably need a clarification here. You need add more properties, like add more building blocks here before, or select properties here. Okay. All right, that's good question. I've never done that before. I was going to say that you can deselect and check the boxes here, and that won't forget the training points, but I'm not sure about adding new building blocks. So I will take note of that, and I will take that for my offline questions, and I will follow up with you by email. Okay. All right. Any other questions? Okay. Good. If we don't have any other questions about the phenologic, then we can continue with the next topic. All right. So now let's talk about the define results. Now that we have the different building blocks, we have segmentation steps, quantification, and also classification steps with phenologic, we can decide in here what are those final results that we want to include in the analysis results. So in here, we will perform any kind of statistical analysis of a population according to the previous building blocks. The purpose of this building block is to generate a table with the final assay readout values for each well of a plate. So you will have this table at the end with those numeric values of your interest. So it could be the case that you calculated all the morphology properties, all the texture properties, but let's say that you just want to see the symmetry, you just want to see the edge texture, for example. So at the end, with the defined results, you can decide what are those final readouts that you want to record in the final results. Okay. All properties that are calculated from previous building blocks can be included in this readout, as well as any combination of formula using this calculation. So if you have any kind of formula that you want to perform with intensity values, you can do that here in the defined results. Also, there are some different methods to generate readout values. So we can specify which method to use for each value. And the results are saved in the database if selected. The results per object could be saved in the database if selected. If you want to have results per well, then you don't need to save all the results per cell or per object. So first we have the first level parameters to define results. So here we need to choose the method. So what is the method that we will use to define those final results? The first method is the list of outputs. In here you will see a list of all the previously calculated properties, and the statistical method can be applied individually for all the properties at once, or you can have different statistical methods for each property. So the list of outputs is if you want to calculate all the properties, or if you want to record in the final results all the properties that you calculated in the building blocks. Now the standard output is to select only one calculated property and the statistical method that will be applied. You can add as many standard outputs as needed, but the difference in here between the list of outputs and a standard output is that the standard output is like a more manual process where you need to choose the properties one by one. And the third one is the formula output. So in here you can enter a formula manually using properties as variables and also formula operators. Okay. The second level parameters are specific to each method. So in the second level, we will set up which properties and which statistical analysis we want to perform for every method. Okay, and then we have the object results. So here we will decide how results per object will be saved in the database. There are three options. We have none, so object results will not be saved in the database to save disk space, so you will get only well results. All will save all object properties calculated by the building blocks in the database. And the third method is use selected well results. So this will save only those properties that you selected in the defined results building block per object. Okay. All right, I see another question. Can you tell which properties ended up driving the A-B regression in the linear classification? So the A-B regression is based on this scatter plot. So if I go back to the scatter plot in these properties that I will show you. Okay, so these are the properties, the relevant properties that the system has identified. So these are the properties that are used in this scatter plot to generate that A-B regression. Okay. Okay, I see another question. In Harmony, meta info is supposed to be added manually in array from form using the assay layout editor. Can we import long form meta information for each well with an important CSV? I think you are talking about the ASCII layout. So let me actually, I will go to the browse just for a moment. Here I have some ASCII layouts. Okay, like here. So if you have a specific info for the different wells, yes, you can import that from CSV from here. I hope that answers the question. If not, let me know, please. Okay. Any other questions? Actually, not sure about what you mean with this square form.
So what we're looking at right here, this is square form.
Oh,
condensed form.
okay, the layout. No, I think here we can
So
only
this
zoom
looks
in.
this looks out like almost identical what we're forced to do in Harmony, where we have all the information for every single one of these tabs that would be defined. You would define here as a meta information as part of the asset layout as information listed in a column on a long form. Like I have it long, not square, and like, does that make sense?
Yeah, I
So
understand what you say, but I don't think I haven't seen that before in Signals Image Artist.
so how do people pull? How do people pull in all their meta information into the asset layout?
Do you have right now like a screenshot so you can send that through the
Yeah,
chat and I can see at it? Maybe
maybe
if I see
let
it.
me get one.
Okay, it's like a list. Can we attach a CSV file with our plate maps instead of entering into the assay layout later editor? I don't think you can attach the CSV file, but you can import the data from the CSV file. But yeah, it's in this form, right? In this it's a layout format that we have in here. But you mean actually you mean like this data, right? Like every label for a specific well. But
Yeah,
you mean like
exactly. So I just attached a
like a list.
screenshot into the
Okay.
chat,
Okay.
and it's literally this layout what you were looking at here, where it's like negative, positive
Mhm.
sample, is defined by one column in this CSV file.
Okay. We could see if you try to, well, my question here is, do you want to see the information in the server on this way, or do you think we can, it's okay if we could just convert the list from this square layout? Because we can try to import the data using that CSV file that you have and
Uh-huh.
see if that will be imported, right? Because it will identify the wells, right? If you have wells on there, yeah, well X, well
Yeah,
Y.
we'll have it has well information included. Sorry, I just grabbed the
Yeah.
screen grab for a bit open. I don't think I
Okay.
scrolled horizontally over to well information, but
All right. Yeah. So we can give it a try to see if maybe we can import the data using that format, and then Signals Image Artist will recognize the well. If it is A1, for example, B2, then it will just put the data in here. But I can again run some tests about that, and I can let you
okay.
know. See
Yeah. Did you want me to attach it to the chat right now, or is that a later
if
option?
you mean the file, the CSV.
Yeah.
That would be great if you think it's not any kind of confidential information. So
Wonderful.
anything that,
Actually, this is very important for us.
Yeah, I mean, because essentially we use this list type information on everything else,
right?
and
Okay.
it was quite a pain to learn that Harmony would not eat it and include it as part of it.
Mhm.
And so we'd have to like come back and include all this in meta information after the fact, and
Right.
that can be a pain because I've exported data off of Harmony for object level, and you guys don't even include well names. It's like well coordinate.
Mhm.
It
Okay.
doesn't make
Yeah, you
like,
mean like to have the metadata attached to a specific well.
yeah.
Okay.
I
Yeah,
mean,
we
it's fine to do it afterwards. It's just that becomes my life afterwards.
The reason why we really bought Signals was we were told it was good for high throughput applications. And so one of the things in high throughput is that we're going to be doing many, many plates with all different components in every plate, right? Because we're screening compound libraries. So we want to be able to attach our compound information to, the image data.
Mhm.
Um, Hopefully at some point we're going to get to the part of Signals where we can do some graphing and statistical analysis of the data. We want to be able to label those data points with the actual information, not just for this was plate 27 well A3. We want to actually know, oh, that was, whatever Gardasil or whatever treatment that we did.
Mhm. Yeah, yeah, totally understand. And single image charts, and actually it's the last topic that we will see in a moment. It can also run those analysis in the batch analysis, so you can perform the analysis in a lot of plates at the same time. So that's true. But I understand that maybe the metadata input, it's important to do it also like in bulk, right? Um, so yeah, actually, if you have any file that I can use that you think the information is something I can use, feel free to
Yeah.
attach it. I can do run some tests and I can let you know.
Oh, I'm going to be attaching it shortly.
Okay, thank you. All right. Do we have any questions before we demonstrate the define final results? Okay. Good. So I will continue here with the image analysis. And now we will see the define results. Oh, sorry, here in the training, I need to accept the training. That is important. Okay. And now let's continue here with the define results. The define results building block will be always put here by default at the end. So this is not a building block that you need to add manually. Automatically, you will have the define results here at the end. Then you can choose the method. So as we saw before in the presentation, we have 3 different methods: list of outputs, standard output, and formula output. If you are interested on having a lot of properties as final results, then we can use the list of outputs. Okay. And here in the list of outputs, if I display here, I will have properties for nuclei. So all the intensity properties, texture properties, morphology properties, right? And also classification properties can be calculated in here. And all you need to do is deciding here if you want to apply a specific statistical method to all the properties. So let's say that I want the mean value to all of them, so that will be applied to all of them. Okay, so this is very quick if you want to calculate all the properties. But if you just want a few of them and you want to manually add one by one, then we need to use the method in here that is called a standard output. Okay, so in the standard output, we will have the property and then the statistical method, right? So if I have, for example, intensity, okay, then here I can decide if that is the mean, standard deviation, and more. Okay, and I can add as many standard outputs as I need, right? So if you want to add properties one by one, you can do that from here. Okay. If you need to remove one of these blocks, what you can do is just click on the method dropdown and then click on the blank space, and that block will be gone. Okay, this one as well. Okay. Actually, if you want, you can send that to my email. I will put here in the presentation at the end my email address, so you can do that on that way. Thank you. Okay. And the third method in here is the formula output. So if you have a specific formula to run in here using the previous calculated properties, then you can define the variables in here, and then you can choose in here what is variable A and what is variable B. Okay. And if you add more variables in here in the formula, so if I have the letter C, for example, the letter C will be added to the variables list, and I can choose in here the property that will be used for the variable. Okay, I will get rid of this. Okay, and I will calculate all of these properties. That is ready, and then we have the object results. So here we need to decide if we want to save all the object results in the system, or if we don't want to save those object results. This will save a lot of disk space. So my recommendation is that if you are not interested on the object results, but you just want to have the well result, then always click on none. So that you just make sure that you save some disk space for those kind of results. Okay. Once that is ready and we have the final building block, we can go ahead and save this analysis sequence to the database or to a local file, depending on what you need. Right, so we can click on this icon over here. I will put here my initials, training analysis, and then click on apply. Okay. If we see here the primary navigation, in the measurement that we have been using, my training analysis has been added to my measurement, right? So that is saved to the database. Now let's see finally how we can run a batch analysis. Now that we have the analysis sequence, how we can apply that sequence to multiple plates, multiple measurements at the same time. Okay. So in the batch analysis, we will have this screen where we can select the measurements. It could be an entire screen, it could be one plate with multiple measurements, or only one measurement. It's completely up to you on how you want to analyze the information here. Something important to take into consideration is that if you use a large screen with a lot of measurements, a lot of plates, take note that it will take longer. So it depends on the size of the data, how long it will take for the analysis to be performed. Okay. And then in here, you need to select the analysis routine. So we need two inputs, the measurement or multiple measurements and the analysis routine that we want to use. Okay, so let's go back to Image Artist, and now let's go here to, to the batch analysis. Okay, this is the icon at the top that we need to select for the batch analysis. Okay, so first we need to select the data. So for that, we need to go to the primary navigation, and in here we can select one measurement, we can select a plate, or we can select even a whole screen. Okay. Since I don't want to take a lot of time running the analysis, I will just select this measurement, just one of them. If you need multiple, then you can select multiple. Something important, because it's not very intuitive, if you select more than one plate like this, and you have here in the table view the data selection, and for some reason you need to deselect one of these measurements, what you need to do is right click on it and then click on deselect. Okay. So that is the data. Then we have the analysis sequence. If you have an analysis sequence as a local file, then in here you can just upload the file, right? But if you have an analysis sequence in the database like this one, my MM training analysis, what you need to do is click on it, and that will be added to my analysis. Okay. I see the question that can you select multiple analysis for one measurement? No, you need to select in here only one analysis routine for all the measurements. So the same analysis routine here will be run for all the data that you choose here in the beginning. If you need different analysis for different measurements, then you will need that. You will need to do that in separate batch analysis. Okay. And then we have the the run analysis in here that we always need to use the default. Okay. And then we just click on start. Okay. It will take some time in here. In this screen, nothing will happen. You see that now the batch analysis is ready for a next run. So easily you can just select another measurement, select another sequence, and run. To see the progress of this batch analysis, we need to go to the job status. So this is where you will see the progress of all the different imports of data and also the batch analysis. Okay, so we'll click on the job status. You see here that right now it is running. You can see the previous duration times that we had for other batch analysis. So for this type of data that is very small because we are using just a measurement, it took 2 minutes, 1 minute and 40 seconds, right? 1 minute and 30 seconds. So it's very quick. And obviously, if you have a real large screen, it will take longer, right? Depending on the amount of data that you have on there. So all we need to do is keep refreshing until the status says complete instead of running.
So, related question on this topic, right? So, we have one analysis sequence to queue up, like say 5 plates of images to analyze, and then we have another analysis sequence that we need to analyze in the same 5 plates. So we can immediately line those up in the next batch job and just queue it, and will it do it simultaneously? Will it do it serially? How will it go?
It will do it. I don't remember right now. It will be simultaneously or seriously. Actually, let's do the test. Let's run another sequence here and see if it does it at the same time or if it waits for the other one to be completed. Actually, this is almost completed. So let's see, batch analysis. Cytoskeleton, and then my sequence. Okay, and run.
And then so like immediately right there, if you so it's pending. Okay, so you can do one batch at a time.
Let's do another one just to see how it looks if I select another one and I click on run. Let's refresh. Well, both are pending right now, but the first one is completed. So we can wait just a few seconds to see when this one starts.
I think you had some message at the bottom on the other tab. If you go back to batch analysis.
No, this is that
sorry.
it is ready for the next run. Yeah,
I just saw the hash key and I thought it was some error message.
Oh. Where? Oh, now you see here. Yeah, so it's seriously, yeah, it's serially. It's first running the first one, and then the next one will be pending until the other one finishes it. Okay,
Right.
good question. Any other questions about this? Okay, so once the batch analysis is ready, we can just refresh the data tree. It's important that we always refresh the tree because we see that right now, probably we don't see the results. So if you don't see the results, we need to refresh. And then here we will have the results. The results will have the same name as the analysis sequence, right? But the icon is different. I believe in here, yeah, you can rename the results. So if for some reason you need to put a different name, you can do that from here. And if we click on the results, we need to go to browse, then we click on the results. We will get all the data in here, the summary per well, and also information about the results. Okay. We can also download these results as a CSV, right? So we can click here on download and export that to our computer if we need to. Any questions about this? Where I see another question in the chat. Where do you refresh? Does it automatically refresh? Okay, to refresh the data tree, we need to click on this icon over here. It is at the left side. It says refresh tree. Sometimes it automatically refresh. If you're talking about the job status, then I just did the refresh of the browser, right? You can always use just the refresh of the browser and you can refresh the job status. And the download button is in here, right? It says download in the summary of the results. But to do that, first we need to select the results that are in here in the data tree and then click on download. Akshay, you had a question?
Yeah. So this is kind of beating a dead horse a little bit, but so we added batch job to the queue, and then it immediately starts running, and then you just keep adding to the running queue, or is it possible to say like, hey, here's the series of jobs I want you to do, build up a queue, and then hit go on something, like because that's kind of how we do it. We were doing it in Meta Express when we'd have like this one a specific analysis for one plate. We add it to the queue, go set up the next plate analysis, add that to the queue, and then we might different analysis, but and then once we build the queue, we just hit run on the whole queue, and then everything starts working on eliminating the queue. But I guess this is sort of a running queue, if that makes
Right.
sense.
Yeah. In this case, everything that you put when you press the button of start in the batch analysis, immediately will be added to the queue. So, and it will start running automatically. The rest of the batch analysis, as you saw, will be pending,
Mm-hmm.
but we don't have a way like to what I'm hearing is that you have like a queue and then you just decide to run the whole queue, right?
Yeah, I mean, essentially, it's where exactly it's very similar to how we set up just now,
Okay.
except nothing gets run until you say run.
Okay.
So like we set up like two batches, right? In Meta Express, we would say set up those two batches, and it'll all be lined up in the queue. And only when we say we would tell one of the worker computers to run, only then does things get pulled off the queue and start processed.
Yeah, or the reason why this is different is all of this is happening on one computer. Like we're all just remoting in over the internet. So there's only one computer is crunching on the numbers, so it can only do one thing at a time. Whereas for our other system, we've got like 4 computers that can all do it at the same time, and so that's how the jobs are being assigned. Even though it seems like we're all at different computers, we're actually all opening the same computer remotely. I'm sure you
Right.
know more
Yeah.
about that than I do, but
Yeah, right.
so if somebody else logs in, like say somebody from the lab logs in and starts running an analysis, it's going to go in the same queue. So whoever, it'll be like first come, first served.
That's correct.
If somebody is, they just started an analysis that's going to take a week, and then you just want to run a thing that takes a minute, I guess, yeah, that's another question. Can we like pause some and let the other one do first? I think we covered it at that last training, but I can't remember the answer.
You mean in the admin training?
Yeah.
Yeah, I'm not sure. From the user interface, I don't think we have that option, but yeah, I can find that out again. Let me make a note. Okay. I saw another question that if you can let's see, what was the question? Automate analysis. I don't think you can do it. Only by clicking on the start button, you can add that to the queue. So we don't have a way to have any kind of an automation for that. Okay. Any other questions?
I'm all done with questions. How about you, Akshar? Aswad?
It's been informative. I'm sure I got one question. Oh, I emailed you the plate map. Can we experiment with that?
Yeah, sure. Let's do.
I'm gonna have to leave, so I won't get to see that part. So I'm really glad we're still recording. And thanks, Akshar, if you don't mind continuing with that while I'm gone. And I just remembered I do have one more question. We didn't actually get to viewing the data. I know Signals Image Artist is also able to do graphs and statistics and z-scores, and will that be a separate training?
Okay. Let's see if this is what you are talking about. Let me just select here results very quick.
In Harmony, it's called an evaluation when you have all these, graphs and things, heat maps.
You mean like this, right? Like we have heat maps, result comparison, pedometer comparison.
Yeah. It looks a bit different than what I was shown when we purchased it. They had some really.
Do you have some specific, specific graphs that I can just take note of that?
PA plots. But it wasn't clear what all they did within the Signals Image Artist environment versus what they had taken the data from here and then analyzed it elsewhere. Yeah, I don't know. So have we reached the limit of pretty much what Signals does?
Yes and no. I mean, this is the basic training, so it could be a lot of things that we didn't have the chance to cover on this training. I know that we have some other trainings for like 3D analysis, cell painting, and more. But if you have any questions about that or interested in specific topics that you didn't see on this training, please reach out to the project manager. I think it's Ray, if I'm not wrong. I know he can provide a lot of information about that better than me.
Yeah, we would like the cell painting training that you're offering. So who does that one?
I think that would be Arturo or another trainer that we have that is called Raquel. But we have that material ready, so we could do that.
Great, awesome. Yeah, we'll take all the training we can get. And I probably for the 3D, I think the ILDES lab might be interested in that.
Okay.
So we'll take all the things that you're offering. I'm gonna need to leave, and I see Akshar's got his thing up. So yeah, I'm really looking forward to see what you guys get accomplished with this. But I'm gonna say goodbye. Thank you so much, Mariana, for being here today and doing this training with us.
All right, no problem. Thank you. Bye.
Bye.
Okay. So Actually, what is the well? This is the
So,
well, right? The well name.
so I essentially have developed a system to overload one CSV to be compatible for multiple various systems. But really, the critical columns here are 384 well, molecule ID or human readable name. So HIJ MN.
Okay. All right. Let me see
Those are the
something.
most critical columns. Obviously, F and A or B are also like useful columns in terms of determining what the plate experiment is, but it's as it seems like we have to lump it with inside a screen or inside a plate. So it becomes moot a little bit.
Mhm. Okay. Let me see. Mm,
Wants you to reload it.
Oh, let's use this one. Okay. So I assume that those columns will be like the layers, right?
Yes.
So
Yeah.
here we'll have like well name and let's do just two, right? Just to test. So we have well name and you said molecule
I
ID.
and J are practically the same thing. So
Okay, so let's do molecule ID. Okay. All right, now let's see what happens if we choose the file. If we ask us to. Hmm. All right, something went wrong, but now this training environment that I have is not like exactly configured the same as you have.
mhm.
So it can be that we have some issues here, but let me try again. So, and if not, I can also reach out to Arturo. I know that he has a lot of experience with this. So in Harmony as well. So let's see.
Yeah, because the thing is, like we got our Harmony training from Robert, and I asked Robert this exact same question,
mhm,
and he's like, no,
mhm. In
or he's like, I
Harmony.
don't. Yeah,
Okay.
ultimately the answer was no in Harmony, but he didn't seem to understand my question exactly, that how each column is a layer, and we can. I just don't have it in square form. I have it in long form.
Hmm. Yeah,
Like
it
what?
seems like it even doesn't let me do the import because of the format, right? So
Yeah. So
initially
what is the
it's
expected format then?
the expected format. I assume that in here will be this, right? That you have columns and rows in the CSV for the different wells and um, sorry, for different, yeah, columns and rows for the wells, and then you just import the values in here. But I know it probably doesn't make any sense because the data usually is in the form of a list for every well and every row. Um, so let's do this. I will take this offline. Actually, I will follow up with Arturo as well to
Mm-hmm.
see if we have a way to convert that to this format, or if definitely the software needs that format instead of the list.
I mean, it's literally just a reformatted thing. I got semi fed up with having to need to do that, so I just started writing one myself, but I haven't finished that.
Mm,
At least
okay.
it would like make a long list into a square block, however you're expecting it, here, and then each one is a.
And you're doing it with Excel or?
No, in Python.
Oh, in Python. Okay.
I was that program of reformatting our converter, that plate map converter. I was starting that in Python. I haven't finished it yet, but that's literally what this is. It's just a plate map converter where each column from the CSV would be converted into this condensed layout, and then we're stuck with copy paste. But we have for each sheet in a workbook.
Right.
It becomes each column becomes a sheet in a workbook because it's kind of bullshit, but that's how it would be. I would say. Also, it has some comments. Yeah. Do you know what that typical CSV that it's expecting?
What do you mean with CSV format? I mean the .csv file, but
Like
I don't know.
structure. Oh,
Oh,
you
you
just
mean,
you assume that it's
okay.
square form, right?
Right. What I'm assuming here, since this is not accepting the CSV that you sent, is that the format should be this same format, right? A square where you have all the different values in the different wells, exactly as you have it in the CSV. I think it will be uploaded here in the layout. I haven't done it before, but I can again do it on my own and see if that's the case, if that's the format that is expected. Because you see that automatically. Oh, automatically, if I choose the filing here, it will just not accept it, right? Because the CSV does not contain a valid assay definition, probably because of the format.
But it looked like when you tried to download the assay definition, it dumped out a XML file.
Yeah, let's see if I can open this. It's a CSV. Mm,
Yeah, it's an XML. Gonna want
Yep.
to open it as a text edit.
yep.
Text editor, not this thing.
Okay, I will play around with it to see if we find out a way to do it. I understand what you want to do, so that's good. And
Yeah.
I'll let you know if it's something that image artists will be capable of doing, or if we need to do some kind of workaround, right?
And if we need to write something for in-house to convert it to XML, because apparently, at least based on what you just downloaded, it's looking like it's expecting an XML. Then I guess
Well,
that's what it'll have to be.
here I think it said, oh, right, yeah. I thought it said import CSV, but you're right, yeah. If it is expecting XML, that would be strange, but yeah, you're right. I will take note of that. See? Okay. Hey, well, anything else?
That's what I had. Looks like I need to continue my work on making this stupidly simple but still annoying
Yeah.
format converter.
Well, good luck with that. Sounds very cool. A bit simple.
It's straightforward, but it's annoying, right? Because like, why can't you just
Mhm.
live with the list?
Yeah, I know that PerkinElmer has some other products, like it is called Inventa. Also, we have this Spotfire that it accepts exactly that format that you have. Um, so maybe we can just I will just research to see if that is something that Signals Image Artists can also accept or not. But I know that we have some other products that accept that format in the format of a list, because it's that is the usual format that you get when you run experiments on a plate.
Yeah, right.
Okay. Well,
As I said,
I've-
it's stupidly inconvenient.
yeah. All right. Do we have anything else? I think
I
that would
think
be it.
that answered that meta question I have that I essentially ask every single one of you guys at every training. So
Yeah. Oh, okay. All right. I will research that for you.
awesome, thank you.
Okay. Thank
Aswad, did
you.
you have any other question or?
All
Cool.
right.
Thank you so very much for your time.
Thank you. Have a great day. Take care.
You too.
Bye.
Bye.